Try as I may, I could never explain what I hear when you don't say a thing. The smile on your face lets me know that you need me. There's a truth in your eyes saying you'll never leave me. The touch of your hand says you'll catch me if ever I fall. You say it best when you say nothing at all. All day long I can hear people talking out loud, but when you hold me near, you can drown out the crowd. Try as they may, they could never define what's being said between your heart and mine. The smile on your face lets me know that you need me. There's a truth in your eyes saying you'll never leave me. The touch of your hand says you'll catch me if ever I fall. When you say nothing at all, yeah, yeah. Oh, the smile on your face lets me know that you need me. There's a truth in your eyes saying you'll never leave me. The touch of your hand says you'll catch me if ever I fall. Oh, you say it best when you say nothing at all. Oh, the smile on your face, the touch of your hand lets me know that you need me, girl. It lets me know that you need me, girl.
One, two, three, four.
When the night has come and the land is dark and the moon is the only light we'll see. No, I won't be afraid. No, I won't be afraid. Just as long as you stand by me. So darling, darling, stand by me. Oh, won't you stand by me? Just as long as you stand by me. If the sky that we look upon should tumble and fall, and the mountains should crumble to the sea, I won't cry, I won't cry. No, I won't shed a tear. Just as long as you stand by me. as long as you stand by me. Whenever you're in trouble, won't you stand by me? Oh, won't you stand by me? Just as long as you stand by me. Oh, darling, just as long as you stand by me.
One, two, three, four.
I found a love for me. Darling, just dive right in and follow my lead. I found a girl, beautiful and sweet. I never knew you were that someone waiting for me. 'Cause we were just kids when we fell in love, not knowing what it was. I will not give you up this time. Darling, just kiss me slow. Your heart is all I own, and in your eyes you're holding mine. Baby, I'm dancing in the dark with you between my arms, barefoot on the grass, listening to our favorite song. When you said you looked a mess, I whispered underneath my breath, but you heard it, darling. You look perfect tonight. I found a woman stronger than anyone I know. She shares my dreams. I hope that someday I'll share her home. And I found a love to carry more than just my secrets, to carry love, carry children of our own. 'Cause we are still kids, but we're so in love, fighting against all odds. I know we'll be alright this time. Darling, just hold my hand. Be my girl, I'll be your man. I see my future in your eyes. And baby, I'm dancing in the dark with you between my arms, barefoot on the grass, listening to our favorite song. When I saw you in that dress, looking so beautiful, I don't deserve this, darling. You look perfect tonight. Baby, I'm dancing in the dark with you between my arms, barefoot on the grass, listening to our favorite song. I have faith darling, you look perfect tonight.
Aku Nana. Percaya nggak? Romantis itu.
One, two, three, four.
Every breath you take and every move you make, every bond you break, every step you take, I'll be watching you. And every single day, and every word you say, every game you play, every night you stay, I'll be watching you. And oh, can't you see, you belong to me, and how my poor heart aches with every step you take, every move you make, and every vow you break, every smile you fake, every claim you stake, I've been watching you. Oh yeah, since you've gone I've been lost without a trace. I dream the night I can only see your face. I look around but it's you I can't replace. I feel so cold and I long for your embrace. I keep crying, baby, baby, please. Oh, 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 oh. Yeah. Oh, can't you see, you belong to me, and how my poor heart aches with every step you take, every move you make, and every vow you break, every smile you fake, every claim you stake, I'll be watching you. Yeah, I'll be watching every breath you take, every move you make, every bond you break. I'll be watching every single day, every word you say, every game you play. I'll be watching every move you make, every vow you break, every smile you fake. I'll be watching you. Oh, I'll be watching you. Oh, oh. The other night, dear, as I lay sleeping, I dreamed I held you in my arms. When I awoke, dear, I was mistaken. So I hung my head and cried. You are my sunshine, my only sunshine. You make me happy when skies are gray. You never know, dear, how much I love you. Please don't take my sunshine away. Always love you and make you happy. If you will only say the same. If you leave me and love another, you'll regret it all someday. You are my sunshine, my only sunshine. You make me happy when skies are gray. You never know, dear, how much I love you. Please don't take my sunshine away. My sunshine, my only sunshine. You make me happy when skies are gray. You never know, dear, how much I love you. Please don't take my sunshine away. Please don't take my sunshine away. No, please don't take my sunshine away. One, two, three, four. Isn't it funny how time seems to slip away so fast? 1 minute you're happy, the other you're sad. But if you give me one more chance to show my love for you, it's true. I'll stand by your side your whole life through. If life is so short, why won't you let me love you before we run out of time? If love is so strong, why won't you take the chance before our time has come? If life is so short, if life is so short, love is a word that explains how I feel for you. When you're in my arms, all my dreams come true. And when you're not around, you can't hardly see these tears that I'm crying out are for you to be with me. If life is so short, why won't you let me love you before we run out of time? If love is so so strong. Why won't you take the chance before our time has come? If life is so short, if life is so short. Love is a word that explains how I feel for you. And when you're in my arms, all my dreams come true. And when you're not around, you can hardly see these tears that I'm crying out are for you to be with me. If life is so short, why won't you let me love you before we run out of time? If love is so strong, why won't you take the chance before our time has come? If life is so short, if life is so short. So short, yeah. If life is so short,
One, two, three, four.
saying I love you, it's not the words I want to hear from you. It's not that I want you not to say, but if you only knew how easy it would be to show me how you feel. More than words is all you have to do to make it real. Then you wouldn't have to say that you love me, 'cause I'd already know. What would you do if my heart was torn in two? More than words to show you feel that your love for me is real. What would you say if I took those words away? Then you couldn't make things new just by saying I love you. La da 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 da. Now that I've tried to talk to you and make you understand, all that you have to do is close your eyes and just reach out your hands and touch me. Hold me close, don't ever let me go. More than words is all sure, then you wouldn't have to say that you love me. 'Cause I'd already know. What would you do if my heart was torn in two? More than words to show you feel that your love for me is real. What would you say if I took those words away? Then you couldn't make things new just by saying I love you. Da-da-da-da-da-da-da-da. More than words. Da-da-da-da-da-da-da. More than words.
One, two, three, four.
Why'd you have to leave so soon? Yeah. Why'd you have to go? And why'd you have to leave me when I needed you the most? 'Cause I don't really know how to tell you without feeling much worse. I know you're in a better place, but it's always gonna hurt. Carry on, give me all the strength I need to carry on. Yeah, yeah. It's been a long day without you, my friend. And I'll tell you all about it when I see you again. 'Cause we've come a long way from where we began. Oh, I'll tell you all about it when I see you again. How do I breathe without you when I'm feeling so cold? And I'll be waiting right here for you till the day you're going home. Carry on and give me all the strength I need to carry on. Yeah, yeah. So let the light guide your way. Hold every memory as you go, and every road you take will always lead you home. Oh yeah. It's been a long day without you, my friend. And I'll tell you all about it when I see you again. 'Cause we've come a long way from where we began. Oh, I'll tell you all about it when I see you again. It's been a long day without you, my friend. And I'll tell you all about it when I see you again. 'Cause we've come a long way from where we began. Oh, I'll tell you all about it when I see you again. Oh, I'll tell you all about it.
2, 3, 4.
You may think that I'm talking foolish. You've heard that I'm wild and I'm free. You may wonder how I can promise you now there's love that I feel for you always will be. You're not just time that I'm killing. I'm no longer one of those guys. As sure as I live, oh, this love that I give is gonna be yours until the day that I die. Oh, baby, I'm gonna love you forever, forever and ever. Amen. As long as old men sit and talk about the weather, as long as older women sit and talk about old men. If you wonder how long I'll be faithful, I'd be happy to tell you again. Oh, darling, I'm gonna love you forever and ever, forever and ever. Amen.
They say time takes its toll on the body, makes a young man's brown hair turn gray. Well, honey, I don't care. I ain't in love with your hair. And if it all fell out, well, I love you anyway.
Oh, they say time takes its toll on a memory, makes people forget things they knew. It's easy to see, oh, it's happening to me, 'cause I've already forgotten every woman but you. Oh, darling, I'm gonna love you forever, forever and ever. Amen. As long as old men sit and talk about the weather,
As long as old women sit and talk about old men.
and if you wonder how long I'll be faithful, oh, just listen to how this song ends. Oh, darling, I'm gonna love you forever and ever, forever and ever, forever and ever, forever and ever. Amen.
Hai pemain satu, siap hadapi tantangan ini?
Hey guys, thank you so much for watching our video, and thank you, Summer, for being a part of our video.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon everyone. Selamat siang semuanya.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, thank you very much for still joining us in summer course for today. So today we will have about two speakers. The first speaker comes from Turkey. His name is how I address is Professor Dr. Mehmet Ulas Sinar, and from Erciyes University, Turkey. And that's the speaker for the first session. And for the second session is Dr. Vincent Guyonet from University of Georgia, Canada, with the topics of trend of poultry industry development in pandemic COVID-19. So after we have the lecture, then after that we will have the post test. According to the schedule, we will have a post test after the lecture in the afternoon, maybe around 5 o'clock. That will be held by Dr. Lucia and Dr. Sri Darwati. So we are still waiting for Professor Dr. Mehmet Ulas Sinar to come.
I'm here.
Oh, I'm here. Oh, thank you very much. Excuse me, sir. How do I address you?
So actually, you can call me as Mehmet.
Mehmet. Okay.
Mehmet,
That's all right with you? And maybe is it all right with you if I call you Professor Mehmet?
You're welcome. So no problem.
Okay. So I have some difficulties for finding your CV in the internet.
Yeah. So yes. So I'm so sorry. So for all participants, actually, I would like to welcome all you. Yep. So I'm sorry. I'm in vacation actually. That's therefore. So I have a limited access to internet. Okay. So I've actually I couldn't share with Dr. Professor Asep with my CV. Actually, he contacted with me so to send him. But this is my fault actually. So, but later on, so I can share with him. Maybe if he has a blog or any kind of website, maybe he can put my CV to that blog actually. But I can shortly introduce myself.
Okay.
So if you allow me actually.
Okay. Please wait a moment. Because from here you have time until Indonesian time about around 2:40 p.m.
Actually, that's a quite long time. Maybe I don't have so that much slides to introduce. So, but after after finish my presentation or talk, so yep. Maybe the participants can ask questions. Then we can, how can I say, go on with like something like dialogue. I mean, okay. So question and reply. Yeah. So okay. this kind of stuff. Yeah. So
okay.
My topic is so. If you allow me, so I would like to introduce myself.
Okay. Please now I would like to have you as our speaker, and the time is yours.
Okay. So can I share my? Yes, I can share. I think my screen as well. Yeah. So dear participants, so ladies and gentlemen, welcome to this session. So I would like to thank the Professor Asep actually organizing and inviting me as a guest speaker and guest lecturer in this session. So my name is Mehmet Ulas Sinar. So I'm working as a full professor at Erciyes University. Erciyes University is one of the first 10 universities in Turkey. This is located in the central Anatolia. Maybe you know, so larger cities from Turkey, so like Istanbul or Ankara, but this is located in Kayseri. It's in the heart of the Anatolia, central Anatolia. So we have around 55,000 students in different aspects, and agriculture is one of them. So I am also working as an adjunct faculty at the Washington State University College of Veterinary Medicine, Department of Microbiology and Pathology. There also I am working about the sheep genetics, so small ruminant genetics there. So today I would like to give a presentation or talk about the genomic selection. which is an important and hot topic in the world at the moment. Actually, so there is a still difference in genomic selection between developed countries and developing countries. So, but developing countries and developed countries actually take a long way about that one, but developing countries still trying to adapt their livestock management or livestock breeding into system. So today we would like to talk about what is genomic selection and how we can utilize this one in the animal breeding. I will try to give you not only theoretical, that knowledge, because theoretical knowledge we know, yeah, so there are a lot of websites or video, that in the YouTube, but so I would like to show you how we can utilize this genomic selection data in practical animal breeding. That's the important part. So because this is not easy to understand with new beginner scientists, so what is genomic selection, how we can utilize this is already spoken in many places, genomic selection, but is it true or is it applicable to the real animal breeding, or is it just a theoretical things? Today we can talk about this kind of stuff, and maybe you at the end of the meeting, so you can ask questions and we can, discuss together to find the right answers. Yeah, so now I would like to share my screen with you. So I don't know if I can be successful because I don't know how to share. This is a Apple.
It should be a green sign.
Yes, green sign, but yeah, since this is not Windows, I don't know how I can share. Still not shared, I think. Let me try. I'm so sorry. Privacy. I need to open system. Sir, I need to change some open system preferences, security grant accesses. So I need to change my I need to change my adjustments actually.
Okay.
So let me try, because it's giving an open system preferences. Security grant access. Look. Yeah, there he is. You can see, I think, yeah, the presentation.
Yes, sir.
Okay. So can you see?
Yes, sir.
Yes, we can.
Okay, good, good. So I would like to give information about our studies, which we did it actually in with Dr. Asep in University of Bonn. So the phenotype is will be meat quality. But firstly, I would like to give some information about what is genomic selection. So let me. So the Mike Goddard and Ben Hayes is if you can see the website of PubMed. So the genomic selection is a form of marker assisted selection in which genetic markers covering the whole genome are used to all quantitative trait loci are in linkage disequilibrium with at least one marker. So this is the generic term of genomic selection, not only in livestock. I mean that this is also for all living things. This is accepted with, or we can assume that this generic term for all, how can I say, for all living things. But nowadays, this is how can I say, this has been changed. Before in 90s, it was, how can I say, accepted as, regarding means that at least one marker. But nowadays, with the development of holistic approaches like SMP chips, which is covering thousands of SMPs, or nowadays, genotyping by sequencing also it's possible, and day by day it is getting cheaper. And so I see some papers relatively new that they are not genotyping animals with SMP chips, rather they are genotyping the animals with sequencing with whole genome sequencing platform like Illumina or PacBio, or new approaches or new instruments are coming to market. So means that we try to understand the phenotype and genotype regression in, or we try to understand the genotype of animals, or not only genotypes, transcriptome or proteome or metabolome. We try to understand in holistic approaches, and we try to merge this information with a relevant trait. And today I would like to give you some example of meat quality trait in pigs. But this meat quality, not only in pigs, you can utilize this information with cattle or chicken or goat or sheep. So means that meat quality is interested in with different livestock species. But I would like to give with our previous studies, which we did in University of Bonn in Germany, with the pig muscle quality or pig meat genetics here. And so I would like to mention you about the introduction of meat quality, genetics of meat quality, and I would like to further on move on another genetics of leg weakness in pigs, or as I said, you can utilize this information in many species, like all livestock species, inverted teeth or immunogenetics, I mean that. So meat quality is a generic term to describe the properties of perceptions of meat. Means that quality is the composite those characteristics that differentiate individual units. These individual units can be in different, how can I say, meat color, pH, or I mean that drip loss, water holding capacity. Many traits are determined in to measure the meat quality. So there are a lot of factors affecting meat quality. Transport means that large time, disease, animal handling, nutrition, post-slaughter handling, pre-slaughter handling. There are a lot of environmental factors affecting the meat quality, but we focus on the genetics. So our aim will try to find which genes or which pathways are related with meat quality, and how we can utilize this data to select animals for better meat quality. So the meat quality traits, the heritability actually starting with if you are working in genomic selection and practical genomic selection in animal, in livestock breeding, you should know about the heritability degree. This is the difference, one of the main difference between developing and developed countries. So because since in developing countries, a recording of animal data I mean that, so the pedigree information, so the sire dam information, the relativeness of the animals, so how they come from in different, you can say hello. Yeah, this is my son. I'm sorry. He would like to say you hello.
Hello.
Halo.
Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam.
Masya Allah.
So there are, as I said, so this is the main difference. If you would like to use the genomic selection, you should know about the heritability of the desired trait. This is must, because if you would like to calculate the genetic gain, you should know about the heritability. But calculating or estimating the heritability degree, it's not easy. Yeah? So there are now new approaches like Bayesian methods, Bayes A, B, C theorems. But for all of this one, to estimate the heritability degree with accurately, so you should know about the pedigree and recording of animals, sire dam information, relative means that brother and sister information. You should know about all these traits or all these recordings to estimate the heritability degree as numerical. But if you don't know about the heritability degree, and if you would like to still use the genomic selection, that's also possible in some aspects. I will show you the example of that one. So, but this is limited. If you, for example, if you would like to do it in a sheep population, if you would like to develop, means that, so progress in meat quality in sheep, so you should know about the pedigree of the sheep to calculate the heritability. Then according to the heritability, you should guess or estimate, you should calculate, I mean that, the genetic gain. So how my marker information can affect my genetic gain generation to generation. To calculate or estimate this one, you should know about the heritability degree. That's why I also always mention to the students, so it's the same problem also in Turkey. Recording or so means that herd book system is not very, how can I say, developed in Turkey as well. And most probably all developing countries is not very well. That's why we cannot calculate the heritability. So in that sense, even if we find the genomic selection, or even if we, how can I say, apply genomic selection, or even if we find a marker, that's for us, it's quite hard to estimate the genetic gain, so generation to generation. And there are some good examples for that one that I will show you during the presentation. So, and as I said, so if you would like to, so genomic selection is in a generic term is to know about the QTS, means that, so the special genome regions that affect the, how can I say, that affect the desired phenotype, like meat quality here. And that's why, so there are some different, how can I say, approaches to estimate or to find QTS. So the first one is the positional QTL, positional one, functional one, or direct biological one. The positional one is depending or based on the mostly in DNA knowledge, DNA information. So means that nowadays, so as I said, so genotyping by sequencing means that genotyping the DNA and finding the variants according to the annotation, means that genome annotation to the reference genomes, and then so estimating the genotypes, genotyping the genotype the animals. And if you use, for example, so I mean that the SMP chips means that, so in 50,000 for example, sheep or pig, or there are 700,000, so 700,000 SMPs also available for cattle, for example. So these are all used to genotype the animals by whole genome methods. And so these are all depending on the positional estimating or finding the positional QTS. So that means that the regression or association between the DNA variants and phenotype. I mean, and the second one is functional. So this could be done with using, for example, omics technologies. Expression means that messenger RNA expression, microRNA expression, large non-coding. There are several approaches at the moment. Since whole genome sequencing is available, measuring the transcriptome is now very easy. So if you isolate the RNA and if you, how can I say, convert it to cDNA with different approaches depending on which platform you are using, actually, like Illumina or Affymetrix or so PacBio. So then you can utilize, you can use, so you can, how can I say, you can understand the whole RNA sequence by sequencing. And if you use this RNA sequence variants or RNA sequence amount, I mean that different between different animals, and you can associate this data also with the desired phenotype, and you can find so which trait is affecting, so means that which trait is affected from which kind of messenger RNA or microRNA or large non-coding RNA. There are a lot of, how can I say, RNA species. So as far as I know, in the literature, we can know that so there are 30 kind of RNA. Yeah. So in the genome, I mean that, and you can associate any kind of this RNA according to your biological hypothesis. You can associate this kind of RNA differences, and you can associate this data with the desired phenotype what we are looking for. And also, so the direct biological, this is most valuable one, but it is very hard, and so it is very costly. That's why so it's lessly chosen or lessly preferable by scientists, like proteins, enzyme, metabolites, or hormones. This is mainly done in the human, actually. I mean that if you have so nowadays there are a lot of approaches to understand the, for example, finding biomarkers, or I mean that biomarker is a kind of QTL as well. Yeah, so it means that finding a genome information, and so how this genome information can be associated, for example, cancer types. Yeah, so as far as I know, for example, in Japan, so they are trying to understand, they are trying to develop, for example, they are trying to develop biomarkers or to find QTLs. I mean that proteins in terms of QTL means that not only in DNA form. Yeah, so we've talked about this one in the beginning. QTL could be in protein or metabolite form also, because these are all coming from the genome information, and they try to find, for example, so genome information or biomarker, trying to find the genomic selection method. I mean that this is kind of genomic selection because I call it like that, for example, pancreatic cancer. And how they would like to detect this one, they would like to, for example, identify a biomarker from urine. And when you, for example, leave your urine in a assay or whatever, yeah, so they would like to guess about your pancreas cancer status or pancreas cancer sustainability, or, for example, if you are resistant or if you are very prone to, for example, pancreatic cancer, and they would like to develop this kind of markers. So nowadays in human, so there are a lot of approaches like that in many countries. Yeah, so they try to find strong biomarkers. So especially now, so pharmaceutical industry is now passing to another era. I mean that, so they are trying to, they are actually not maybe totally giving up, but so they are passing to so genome information or personalized medicine. I mean that, so they try to develop therapeutics, so pharmaceuticals according to your genome information. So that's why this genomic selection not only used for animals, so we can utilize also this data with the human approaches. Yeah, so this is just based on, so what is your hypothesis, what you would like to find, what you would like to, which trait you would like to make a better way, I mean that. But so in all cases, we are trying to find candidate genes. So it means that candidate genes, or nowadays, this is a, I mean that old term. Now we would like to find a candidate SNPs, or yeah, especially if you are talking about proteins or enzymes. Yeah, we are trying to still find the candidate genes. So it means that which metabolite, this metabolite is coming from which gene. So this is still, how can I say, important. So sorry about that. And so, yeah, if after you identify these candidate genes, so we should do for association or functional analysis to prove that, I mean that this is really the right gene that makes a that makes, I mean, that improvement in that trait. So you should validate in, for example, if you identify, so if you are, for example, so in a human or in livestock breeding, so if you identify a candidate gene in different, means that positional, functional, or direct biological approaches, for example, in an Indonesian sheep breed or Indonesian human population, then you should validate these candidate genes or mutations, I mean that in different animal species as well. Not only, for example, Indonesian sheep breeds, you should test that one. So is that the real mutation, or this is really, how can I, strong mutation? So you should test this one, for example, in the US sheep breeds or Chinese sheep breeds or in European sheep breeds. So, or also for, this is the same acceptable for human cases, I mean that. So means that if how we can move from beyond the genome, so it means that we are starting from genotyping, the all DNA sequence and variants. This is, I mentioned to you, this is nowadays very easy. So the sequencing technologies is very developed. So in different, by different companies or approaches, but this will not stop, I think. This will be developed also in coming days. New platforms or new sequencing techniques are going to be launched. And then, so we will see in coming days this one. Transcriptome the same. So nowadays, so RNA sequencing is very popular and getting cheaper and cheaper. So that's why, so I understanding the transcriptome of animals nowadays, even for transcriptome before, for example, 10 years before or 15 years before. So the chips, RNA chips, so transcriptome chips were using, but nowadays almost they are not used, and the RNA sequencing is very popular method for understanding the transcript between, how can I say, different phenotypes or whatever the cases you would like to measure. And proteome, almost the same, but still expensive, not easy. Mass spectrophotometer, mass specs, I mean that, mass specs. So they are not so cheap technologies to understand the holistic protein approach. So most of you, maybe you use about the Western blot, means that any single protein you can measure, but proteome, I mean that whole proteomic approaches, so it's still not very cheap, but still used. So if you have a strong phenotype and if you would like to find a really strong marker, that you should go on with the proteome. And metabolome is almost the same. So metabolites are the end products for, I mean that for biological organisms, and all kind of, how can I say, phenotype occurs after metabolome. And this is all called the system biology. And so, as I said, genomic selection is also very strongly associated with system biology. You can, so although we try to find the markers for one of these ones, so for example, not every lab is not very successful to work with system biology, but I mean that different part of the world, the labs are taking this information from, for example, somebody produced the data for genomic selection, for example, from the genotype data, and one of the Indonesian lab is getting, for example, the transcriptome, use this knowledge and validate this one in the transcriptome level, for example, and then if you publish that results, one of the Chinese lab or European lab get this data, and they are going to continue sometimes in protein level. Yeah, so that's why this is step by step, and so block by block. So we try to build a strong wall, so all about the scientists in the world, and we use our, for example, paper data, published results, or this kind of stuff, and we put a little bit step forward to produce a strong, how can I say, produce a strong marker for relevant trait in animal breeding. And but some labs are very, how can I say, they have a lot of money, I mean that, no budget problem. They can work in the system biology, especially nowadays the Chinese labs are very, how can I say, very skillful or talentful to work in system biology, and they are publishing a lot of papers regarding that one. So the workflow of that work is actually, this is the animal, I mean that, so DUPI, Duroc and P-Train, so crossbreds and a commercial population. So it was 200 animals that time. Nowadays, so genomic selection is done with, for example, dairy cattle population, thousands of animals. So 10,000 or more animals is used for example, genotyping and understanding, applying the especially genomic selection in US and Europe, as far as I know. So the German Holstein population, they genotype 10,000 cattle last years and try to understand the genotype phenotype regression in 10,000 animal scale, actually, with their whole pedigree information, knowing the heritability, everything, and they are calculating their genetic gain very accurately, I mean that. So how we can do that one? So SMP chip genotyping, this is trying, so this is a positional QTL, understanding the positional QTL. If you can see here, for example, this is the genome information and trying to strong SNPs regarding meat quality traits here. And then, so the functional ones is coming from the whole genome gene expression. So, or this is the old method, as I said, the gene chip method, but nowadays, so as you can see, RNA sequencing is also used. And then the proteomics we did that time. So in a small scale, I mean that. So if you would like to genotype 10,000 of animal in proteomic level, it is almost impossible because it's really, really expensive. But with small number of animals, it was possible. And we did the whole proteomics, means that whole proteome information about the animals. And we try to understand the candidate proteins, which are associated with our traits also. And during this time, so not only genetic information is important, genomic selection is not only associated with genomic information, it is also, genomic selection is also associated with epigenetic regulations. Especially, this is coming, already actually a lot of papers or research on that one, but there was no practical application on that one. For example, nutrigenomics. So means that how nutrition and genomic information, how it could be integrated. So for example, so because since animal feeding or animal nutrition is a strong environmental factor, so how it is affecting the genome of the how can I say, animal. And this is also, the question is coming, so animals can be selected according to their epigenetic modifications or epigenetic variation, I mean that. This is another question, is coming in maybe in 5 years or 10 years, it will be possible. We will not select also animals only according to their genome information. We will select also animals according to their epigenetic modifications. Now, this is ongoing, this epigenetic information is ongoing on research scale, but so most probably in coming years, it will be included into selection, how can I say, selection methods. And so most probably maybe the US Dairy Cattle Association or US Dairy Cattle Breeders, they are going to starting to ask for example, what will be our nutrition and genotype interaction, how we can utilize this data. Is there any variation? Because we know there is a variation among animals according to their epigenetic, how can I say, epigenetic status. And so this is most probably associated with their also phenotypes. But this is coming question in maybe in 5 to 10 years. So we try to find which genes. So genomic selection is, as I said, the question is which genes or which SNPs or which messenger RNA or which protein. So this, but name generally means that which genes. Yeah. So we try to find the genes for improving the animal quality. So as I said, this is the example. So muscle pH. Muscle pH is most probably you know, all of you. So since you are interested in or working with the animal breeding, so muscle pH is one of the most important characteristics or traits among meat type animals. I mean that, and the pH is affecting the, so as I said, the meat quality traits, like for example, higher, more basic, I mean that more basic basic status of meat, like 6.2 is causing, for example, darker meat, and more acidic one is causing to, so it's called PSC. It's a very pale type of meat, and this pH is also not only affecting the color, it's affecting many attributes, means that like chewing ability, like tenderness, or for example, the water holding capacity or drip loss means that the pale meat, acidic meat is, for example, losing more water, and this water is very important because, as you can see, the vitamins and minerals are washed through this water, and it's losing means that meat is losing. If meat is more acidic, it is losing its, how can I say, vitamin and minerals through with excess water actually. Yeah, so that's why animals is showing variation among this water that lost through, how can I say, after post-mortem. So post-mortem storage conditions, I mean that. So that's why if animals are showing variation, then how we can utilize, how can I say, find genomics selection criteria to make the animals optimal water loss. Yeah, so this was our aim. You see, this is drip loss, how it is measured in the laboratory conditions. We just put one piece of meat, so around 200 grams. So, and we weighted that one into weight before putting into the bag. And then later on, we put into 48 hours kept in 4 degrees, and we measure the meat weight after 48 hours, and we try to estimate. There is a calculation algorithm how you can estimate the water loss, and you can see. So if you go to butcher or grocery, you can see this water loss also in practical conditions. Yeah. So this is the how can I say? As I said, so there are a lot of meat quality traits, not only drip loss. So different parts of carcass, you can measure the different meat quality traits, but this is in detail. You can see how it could be measured actually. So this is requiring a really optimized testing conditions. So it's still, this is, as I said, this is the problem of developing countries. So even including in Turkey, so we don't have this kind of testing, how can I say, facilities. So for each slaughterhouse, that's why we cannot work with meat quality traits in terms of genomic selection actually in Turkey. But in Germany, very good. So especially for pigs, there is very nice herd book starting from farm, and it's ending in the slaughterhouse actually. All animal information you can find in details starting from farm, when they are piglet and when they go to slaughter. So you can find this information for most of each animal, I can say that. So this is, as I said, drip loss, how we can measure determination of drip loss, the bag method, and there is another method. So it's called easy drip loss. So as I said, this is the easy drip loss, not 200 grams, so getting smaller amount of meat and putting these plastic bags. And so since it is the drip loss is a percentage, so it doesn't matter it is 200 grams or 50 grams. So you can guess, estimate, calculate, how can I say, the drip loss in percentage, I mean that. So there are also towing loss. So you can how can I say, observe this one when you are cooking a meeting. So the meat is giving its water into the can, or you can say that inside where you cook the meat, you can find this water in different amounts. In each kind of cooking, you can see this different amount of water. This is coming, for example, this information is coming from, or this observation is coming from the animal's DNA information actually. The genomic knowledge of animal is affecting the cooking loss phenotype. So this is the shear force, as I said. So pH is not only affecting so meat color is also affecting the tenderness or chewing ability of the meat. This could be measured by a special instrument. So it's cutting and measuring the force of the meat resistance, fiber resistance, as far as I know. So the genetic analysis. So this is the what we use we start for QT analysis. These are the old information. So this is whole genome scan. This information is coming from, for example, the microsatellite genotyping. But microsatellite genotyping, most probably, so nobody is using at the moment. maybe some plant scientists are using, I don't know, but in animal information, if you would like to apply genomic selection, nowadays nobody is genotyping animals with microsatellite markers. As far as I said, even gene chips, so this 50K or 700K chips are still used, but so in coming days, most probably it will be, how can I say, left. Yeah, so because the whole genome sequencing is very popular nowadays, still relatively expensive, but maybe in 5 or 10 years, it will be very, very cheap. So that's why maybe we are going to give up using these gene chips, but this is quite old information. And gene expression analysis, so as I said, so this Affymetrix gene chip used for estimating the animal's transcriptome, and this was my PhD study, EQTL study. So this is based on, as I said, so the genomic selection is not depending only in DNA information. So most probably all of you or most of you is guessing which we talk. So genomic selection is based about the DNA data. No, so as I said, so maybe 3 or 4 times I said this one. The gene expression data. So gene expression is showing the messenger RNA expression is also showing variation among animal population. Means that one animal's so among in one, for example, just assume 10 animals and 10 animals, assume that one marker messenger RNA, this 10 animals messenger RNA amount is not the same. So it is showing variation. So, and if you measure 100 animals, you will show this measure variation so more accurately. And this is also associated with DNA information, means that one animal's messenger RNA amount is also depending the DNA information of the animal, and this could be used as a marker also. Yeah, so that's why so genomic selection, as I said, so it's not a easy issue. Yeah, so and yeah, most probably you know about that one. So Jack Deckers, so from the Iowa State University, they have so this Jack Deckers and his group, so all Iowa State scientists, I mean that, so they are very good in livestock breeding on genetics, I mean that. So he published one paper, and so although there are a lot of effort, lot of research, lot of money put on the livestock breeding and genetics, only small number of markers could be developed up to now. Yeah, so this is showing us. So this is not, there is no easy issue. This genomic selection is not an easy issue. It's not only depending on the DNA information. The DNA information, yeah, in some traits maybe we are lucky because, like for example, so DGAT, maybe most of you know about that one. So DG80. So it is double muscling MIF5 mutation, for example. These are the single SNPs affecting the most or explaining the most of the variation. For example, DGAT is explaining the variation in, for example, in meat, milk fat or milk amount of the dairy cattle, or MIF is affecting whether it's a double muscling. So these are the very luckily found, but these are only, for example, I know that I remember the Jack Deckers's paper up to now. So around 30 or 40 markers developed, DNA markers developed for a very accurately directly related with a I mean that phenotype in livestock. But if you assume that there is a 25 or 30,000 genes in one livestock species in the whole genome, this is very small number. And if you assume that there are a lot of traits, this is very small number. So that's why, as I said, it's a composite. Genomic selection is a composite term, and this composite term is not only only made up from DNA information. So the DNA information is used because it is cheapest for analysis. Still, the transcriptome and proteome or metabolome is still so relatively expensive compared to DNA analysis. And DNA analysis could be very easy. So for even so understanding the genotype, the information of the animals. That's why we are still a lot of ways to find right genomic selection criteria for livestock species. This is the project of DRIP. I mean that starting from these old papers, so the genome scan, so means that the QTL analysis, these are all depending on the, so as I said, microsatellite information, but later on we developed this one. So with the, as I said, chip genotyping, maybe the new scientists also going to continue this kind of research with, as I said, DNA sequencing, whole genome sequencing methods. So as I said, quantitative trait loci is a term, so very, very close to genomic selection, very, how can I say, stick on the, so quantitative trait loci term is very stick on the genomic selection term also. So almost we can say that Mike Goddard and Ben Hayes is also mentioning in that paper which I showed you. So quantitative trait loci is almost equal to genomic selection. That's why so we try to find QTLs. So it means that if you have a marker, for example, and if your QTL is so between two marker, you use linkage disequilibrium data to estimate this marker, or you estimate this QTL, how it is associated with the respective trait like meat quality or dairy traits or any kind of traits. As I said, so there are, this was microsatellite data. It was used 127 genetic markers, but nowadays nobody is using this information. What this is, as I said, so the starting point of the genomic selection is all type of genomic selection. As I said, so this is even the comparative maps. Maybe I am mentioning about the history also. So these are the comparative maps because nowadays all genome data is found in the ensemble or NCBI, all species like sheep or cattle or pig or chicken, you can find all genome positions or gene positions. So not like maybe in human or rat or mouse, but for example, cattle is more developed compared to sheep, or pig is also as far as almost the same content with the cattle, but chicken maybe is also good, but for example, sheep or goat is the less developed compared to other livestock species. So, but almost their positions are clear, and these are day by day developed. But before that, so since pig was not known, for example, or for example, cattle was not known, so we compare this information with the human data. So there were comparative maps. So the comparing the positions or QTLs according to, for example, human genome information. So expression profiling for meat quality traits. So as I said, this was the new methods. So we measured gene chip platforms, and it was really so accurate that time. So 23,000 messenger RNA oligos were located on the chip and almost covering the whole genome. Nowadays, as I said, it was almost done and almost given up. Yeah, so the RNA sequencing is very accurate and very, how can I say, popular method for that one. And that time, as I said, gene chip was used to identify the transcriptome of analysis. And so we use divergent phenotype for that one because, as you see, so the transcriptome is depending on the divergency. I mean that. So high pH, low pH, the differentiating genes. The hypothesis was, so what are the genes differentiating between high and low pH is affecting the phenotype. So this was the idea of transcriptome, mostly in transcriptome analysis. So it's like something like a case and control. You have a case story and you just compare the transcriptome with the control study. As I said, so eQTL maybe some of you know, maybe some of you doesn't know, but eQTL is, as I said, depending on the messenger RNA expression is also means that one gene's transcriptome means that messenger RNA amount is also affecting from the genomic selection, can be affected from genomic selection. If you would like to enrich the one messenger RNases, for example, so amount, or if you would like to optimize the messenger RNA content of one animal, so means that you would like to, we should, how can I say, we can apply genomic selection for this messenger RNA content. But this is another approach. But as far as I said, so genomic selection not only depending on the DNA information, it is depending on many levels, many layers, I mean that. So we identify the differentially expressed genes with transcriptome analysis depending on chip data. And then so we applied, as I said, so we applied genome-wide association. So these are after microsatellite genotyping. So that time, it was almost 10 years ago, the gene chips were used, still used. So we are genotyping the animals with, for example, porcine was 60K. Nowadays, sheep, for example, 50K is available, or for cattle, 700,000, 700K chips were also available. Still, so animals are genotyped with this kind of chips. So to understand the whole genome, genotyping, but as I said, so since the whole genome sequencing methods are going to be developed, these were going to be abandoned. Yeah. So in 10 years, most probably these chips, unless they are developed with new technologies or they are cheap, they are not going to be used. But as I said, so these are all if you do, for example, DNA sequencing or chip sequencing, the main aim is understanding the whole genome variation, genotype variation of animals. I mean that. These are the main aim. And after this genotyping, so we identify the candidate genes. So for example, in chromosome 2, so SOC6, NEL1, so or chromosome, I don't know, 1, 2, 3, 4, 5, 6 in SHANK1. So we identified, so genome-wide significantly some, or nebulin, for example, as far as I know that time. So nebulin was the important, one of the important genes. These are the, so DNA markers. Is it associated with drip loss phenotype in livestock? These are the DNA markers. But is it enough? It is not enough, because we should validate this one, these SNPs or these genes by transcriptome or by proteome, whether they are really good marker for selecting animals for drip loss. And so this is the enrichment, or you means that so identifying the function of genes. So after you identify so the genes, like according to their statistical power, so then you should understand so the genes are whether really interested in, or they are associated with your, how can I say, phenotype. In that term, for example, since it's meat quality, for example, NAL1 is associated with calcium ion binding. So calcium metabolism is very important for muscle attributes, and it's highly affecting the meat quality traits, the calcium. Or they are, for example, SOC6 is related with muscle development, or for example, nebulin is associated with muscle development. That's you can find from the gene enrichment websites. Nowadays there are a lot of gene enrichment tools, or you can find a lot of data in the databases like Ensemble, NCBI, or there are some private tools that are developed day by day, developed by different scientists, associating your gene and which function it has, and it could be associated with your prospective trait which you are investigating. As I said, expression QTL, another layer of genomic selection that could be, if you have a very, as I said, maybe in livestock, could be this one very luxury, because always it's not possible to measure the one expression with accurately, or you cannot get the tissue or whatever the biological material sample to measure the gene expression, but is just keep in mind that this expression QTL is another layer of genomic selection. And then, so as well, so we identify genes when we started to publish, as I said. So, for example, this was my PhD paper. So the AMBP, GC, and this PPV1R3B genes are associated with meat quality traits in terms of eQTL. So, and other genes like Nebulin, SOC6, or other genes which I show you according to the DNA variants also published in that one, means that, so we have DNA and transcriptome levels up to now. And then, so we moved on to the proteomic level. So we completed the genetic, so means that the DNA level, and we completed the transcriptome level, and we move on the proteomic level. And we did this one in 100 animals, means that high, so 21, so high drip loss, 21 with low drip loss. And we try to understand which proteins are differentiating between these two groups. And that time there were isotope labeling, so mass spectrophotometer techniques that I don't know in details actually. So, but nowadays maybe different proteome approaches and mass spectrophotometers, mass specs are developed. Most probably, I don't know. So, because long time I haven't worked with the proteomics. As I said, it's quite costly, still costly. That's why it's not easy to work with any kind of lab. So it requires a big project. I mean, that international projects or maybe so governmentally funded big project, because it's very costly analysis. But as I said, if you would like to apply really genomic selection, you should identify, so you should go on with the DNA level to the proteome level, even the metabolome level. So this is the way of how we can. And after this proteome analysis, we like messenger RNA expression, we identify the proteins, for example, TPM1, TPM2, TNNC2. So, and then again, we try to understand their function according to their functionality. Like so, we kick pathways or ensemble or ACBI, lot of tools identifying the function of that genes. So we identified that one, and then so I will show you one paper about that one. So where it is. I'm sorry. Yeah, that one. So we make another publication. So the integrative analysis of metabolic. This was the end paper of that project. Actually, very valuable. I suggest you to, if you are interested in analysis of genetic traits in livestock, I suggest you to read this paper, because this was the, so we make individual analysis from DNA, we make individual analysis from messenger RNA, we did individual analysis proteome, but then we merge all this data together in silico, means that, so with computer tools, so with bioinformatic tools, we merge all this data and we came into one gene. So this is most probably given in the name. It would be NANK. Yeah, this is like, for example, phosphoglycerate mutase. So PCM2, so means that on chromosome 18, because chromosome 18 was in, even if so more than around 20 years before, our group found that chromosome 18 is very important in muscle properties. So in drip loss phenotypes, I mean that, and we found that one. And by this way, we proved that the phosphoglycerate mutase 2 could be used to improve the drip loss phenotype. So in pig breeding, I mean that. So. But as I said, this is an example. This, you can utilize this information, or you can utilize this workflow by identifying in other species as well, like so sheep or cattle or chicken or goat. You can find any kind of, you can utilize this data, any kind of phenotype in different species. But as I said, this is coming from the DNA data. The valuability is coming from that one. So maybe this is a very small picture, but this is the graphical abstract. I mean that. So it is coming from genome, so transcriptome, proteome, metabolome, and phenotype. All this data merged together, and then it's found that PCA2 is very good candidate for I mean that for drip loss phenotypes, improving or selecting the animals according to their drip loss attributes. This is very good example of how this genome data or genomic selection could be utilized in one livestock species. But as far as I say you, so I will how can I say move you? I will show you for other examples as well. So this is an epistatic approach. Epistatic, as I said, like epigenetics. So epistacy is also another layer of, I mean that genome variation. So epistatic effects also is very important, as I said. So genomic selection is so we try to associate with one marker with the relevant phenotype, but it's always not. Yeah. So I can show you. So maybe so for example, so there is a QTL, and this QTL. So there is a QTL on chromosome 1. And this QTL is affecting chromosome 2, and this QTL is affecting chromosome 17, and this QTL is affecting so on. Means that not only, so if you identify even if one QTL, this is not only affecting the phenotype by itself, so it is affecting the phenotype with neighborhood or other genome, genomic or other with other QTLs. So, and this is the actually how we can produce gene maps. Yeah. So as I most probably you are using, most of the labs are using so pathway analysis. So there are a lot of so very, how can I say, complex nowadays. The tools are so very developed on that one. So they are making how can I say a pathway analysis or gene enrichment analysis, and how the genes are affecting each other to influence one phenotype. Yeah, so that's why. So as I said, so maybe Jack Deckers was right. So we spent a lot of money, time, a lot of people, thousands of people are working in the world with livestock species to find a reliable marker, but up to now we find very low number of markers. This was the reason, maybe because the traits are very complex and not only affecting by one marker or QTL. So it is affected by many markers among the genome. And so that's why so lately or nowadays, so people who are working in advanced breeding or advanced genetics, they give up starting to find a single markers. They try to find a so genome maps. I mean that how the genetic means that genes are interacting each other and how they could come together and affecting one phenotype. And they are trying to estimate the percentage of how, for example, there is a QTL and how much that QTL affecting that phenotype. And so by it is interacting with, for example, so if assume that this is the explaining the 10% of that variation. So for drip loss, for example, and this is affecting, for example, 2%. And so by improving only by selecting animals according to only this QTL will with limited amount, it will affect the drip loss. This is also affecting, and this is also affecting. That's why we try to understand the system by like whole genome, not only with genomic selection depending on one marker. I mean that these are the this was the paper of epistatic QTL pairs. So this is not that much focused at the moment. Actually, I don't see a lot of papers about epistatic QTL pairs at the moment, but this is an important part of the, how can I say, livestock genome. And maybe you can work about the epistatic QTL pairs, not only single QTLs, or so I mean that like GWAS, so the QTLs distributed in the among the genome. And rather than that one, maybe you can investigate on how their interaction, so how they are interact and make a so drip loss or any kind of animal trait, for example. So as I said, system genetics, network concepts. As I said, so single QTL is nearly so not ending. I mean that. So there are a lot of papers are publishing so with single QTLs like GWAS or still very popular. But the main idea is how these QTLs are coming together and making that phenotype. And you should draw the maps, or you should like pathways. You should draw pathways. You should explain by this one with pathways. So that's why this is very important. I mean that. So these are the muscle transcriptome network modules. Means that how this this is depending on the correlation of expression. So the close, highly correlated gene expressions are coming together, and they make like modules. So we call it module QTL. So the highly correlated ones brought together to make to explain the module QTL. And this was, as I said, so the 12 QTL module QTLs are, for example, the 512 genes, for example. So their correlation, the expression correlation is very high, and that's why they brought together, they grouped together. And for example, 1,247 genes come together, and so their correlation is very high, and that's why so they brought together and make another, how can I say, another group. And then, as I said, like DNA or transcriptome, and after that you identify the module QTLs. You should identify their functionality, like intracellular signaling pathway and whatever. Yeah, so you should associate with your trait. And module trait relationship, these are the correlation coefficients. And so there is a legend, for example, high correlations in positive way, or high correlation in negative way. This is the legend, how you can calculate that ones. And nowadays, so the R tools are doing this kind of analysis very easily. So there are statistical packages in the R program, and you should, how can I say, open that one, and you can, if you have the, for example, gene expression data, you can utilize whole genome gene expression data with these R modules, and you can calculate and draw these pathways or whatever. And as I said, module QTL, MQTL detection, not only, it is not depending on, as I said, not only one QTL. So as you see, there are hundreds of genes together coming making one QTL. Yeah, so that's why, as I give again, Mr. Professor Deckers's example. So there are a lot of effort has been done, but limited number of markers has been found. So single marker, I mean that. That's why, so as you see, so hundreds of genes are related with respected phenotype. Yeah, so these are the example. This is Bohr taint, Professor Assef's, this was the Professor Assef's project actually. Bohr taint, another kind of trait like Driplos. So Bohr taint is maybe, so Professor Assef is working with nowadays with the Mouton odor. Mouton odor is almost the same with Bohr taint. So I don't know in details actually, but it's kind of a metabolite. Yeah. So, and this, since it is a metabolite, it's done by the genome information. So that's why. So we try to find, and especially in European Council, they say that we are going to ban the castration, because to avoid this taint in pigs, the only way is castration. And since this is against animal welfare, they say that the European Commission decided that, so we are going to ban the castration. And that's why, so to reduce the taint, find another way. So they find, for example, chemical castration, or kind of, so couple of, how can I say, methods for avoiding the taint from the pig meat. But the best way is, so they decided that genomic selection, and then they started one project to understand which genes are causing the taint metabolism or taint formation in the pigs. I mean that. And if we identify a marker related with taint, so then we can select animals against this taint, and we could reduce the amount of androstenol or scatol formation of animals. So because, as I said, so the Bohr taint is so depending on two metabolites like androstenol or scatol. Androstenol is as a male hormone, And skatol is a compound that is done in the gut, in gastrointestinal system of the pigs. I mean that is done by kind of a bacteria, and then it's accumulated. This odor is mixed, so means that goes through the blood system, and through the blood system it's passing through the liver, and after that it's accumulated in the fat. And when you cook this fat together with the meat, so the odor is passing through the another formation that the human can identify. That's why and they say that I didn't test, but so they say that it is very bad smell actually, and consumers doesn't want to feel this smell, and that's why the pigs, all male piglets, are castrated in 7 days old throughout the world. Yeah, so when you think about millions of male piglets, so this is very, how can I say, bad situation. Yeah, so the castration is also occurring this immune problems, and some of the piglets can be died so due to infection after castration. Yeah, so and that's why they say that so we are going to ban this castration and try to find a marker, gene marker for that one. And then, as I said, so genomic selection for boar taint project, and so these were the androstenone levels of 3 animals. As I said, so 3 animal has different androstenone level, and this is caused by, we thought about that this could be caused about the SMP information during the genome. And we make this experiment by using RNA sequencing, for example, and we published 2 or 3 papers, and we try to identify markers. These were the strong markers, for example, related with boar taint formation. And even that with transcriptome means that sequencing, RNA sequencer, you can identify which allele is related with what kind of, for example, so the alternate A, our animals is carrying A, and A is causing, for example, low androstenone, and then that you can select animals according to the A SMP. So this is the basic of genomic selection. As I said, so not only single mutation, so the how these mutations or genes are interacting each other. So we identify that one pathway based analysis of genes. So this is more valuable data, because as I said, genes are not only interacting, genes are not acting by themselves, they are interacting to make a phenotype together. As I said, in the module QTL formation, so it's making so hundreds of genes coming together and make this phenotype. Yeah, so and so after this, I mean that module or single QTL analysis, so we found that FMO, CYP, ESR1, PLIN2, SUT, 2A1 genes are the really good candidates for boar taint selection. And then so we make this we make validation study in a larger population, and we saw that so it's strongly associated. Yeah, so as I said, this is Professor Decker's publication, so it's not new, but you can read about the idea. So he's saying that so we are working too many scientists, we are funding too many money, so but we are finding so low number of single markers. Yeah, so that's why I said that he says that so we have a lot of ways to go on. Yeah, so genomic selection is not easy. These are the, as I said, so single markers affecting a limited number of phenotype. But when you think about the livestock, so there are a lot of phenotypes to be improved, not only for example, not only meat quality or milk quality, there are a lot of phenotypes, especially in these days, animal welfare is very concerned. So there are a lot of phenotypes is coming, or there are papers are publishing about new type of phenotypes. So not classical phenotypes are very well known, but they say that so the phenotype number is also improving with improved technology with in animal breeding or animal husbandry. That's why new phenotypes are coming. So, but the number of markers are not developed relatively. So still they are very, 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 how can I say, limited. Yeah. So this is the hard point of how can I say, genomic selection. But whatever the method, sequencing, RNA sequencing, DNA sequencing, proteome, metabolome, whatever, the phenotype is the king. So you should know, if you would like to apply genomic selection, you should know about the phenotype. You should have very good herd books. Yeah. So means that showing the phenotype information, so sire dam information, grandparent information, so sibling information, half sibs, full sibs. So these kind of informations are very valuable, because so if you would like to apply the genomic selection, you should know about the heritability degree, which we mentioned in the beginning of the presentation. So this is very, very important actually. So as I said, so this was a lot of money is also spent for that one. So, and a lot of people work in that kind of projects. Yeah. So everywhere in Indonesia as well, in China, in Europe, in United States, everywhere in the world. So spending a lot of money, and a lot of scientists are involving in this kind of genomic selection projects. So we appreciate all, but this was mainly done in University of Bonn and Farm Animal Biology Institute in Dummersdorf in Germany, especially Professor Schellender. Now he's emeritus, but Professor Klaus Wimmers also, they are very good animal geneticists in this how can I say, in this area, and I suggest you to read their publications. And so I would like to show you lastly, so I would like to show you some I mean that not theoretical information, so the real, I mean that information. So this is a good paper. I suggest you to read. So the genomic selection in dairy cattle, the USDA experience. This is the paper. Maybe most of you maybe saw this paper. So maybe some of you has not. So this is the paper that explains the 10 years path of USDA's, means that United States Department of Agriculture's in dairy cattle genomic selection project, and how they make the progress by using genomic selection approaches. I mean that. So I opened that publication, but maybe it was most probably I lost. I would like to show you the graph actually. I can open you again. Yes, this is the whole paper. So the genomic selection in dairy cattle. So, and there is one impressive graph here. So this paper is very good. I suggest you to read actually, describing the how the genomic selection is very useful and how it could be applicable in a herd level. Yeah, so it's showing that one, and there is very nice graph for that one. So at the end of the, I think, yes, this is. So for example, between 2000 to 2015, so where the genomic selection has been applied in the US Holstein bulls, so the average gain per year is increased year by year actually. So in first 5 years, so it was around $20 average gain per bull. So then it was increased to nearly $85 per year. This was done by the genomic selection information. So they applied genomic selection, and then here in the conclusion they say that they doubled the genetic. How can I say? There was a nice wording here. So genomics increase genomics means that they selection compared to phenotypic selection. Actually, they doubled their gain. But how they calculate this gain? Actually, they calculate this gain because they know the heritability degree of the animals. They are estimating year by year, or each couple of years they are estimating their heritability degrees in many traits. And this is very, how can I say, impressive, requires impressive workbook recording system. Yeah, so that's why this paper is very valuable and showing the not only theoretical part of genomic selection, it is showing the practical, describing the practical selection, practical part, I mean that. So maybe this is for US case, but maybe you can find this one in European case also. So the German Holsteins, I don't know the paper about in European case, but most probably there is. Yeah, so you can find this kind of papers and you can read so how this genomic information can be utilized, and so how this as you see here, not only so one phenotype is tested, so they are recording a lot of phenotypes, so and they are making their genomic selection not only based on, for example, milk yield or milk fat. So yeah, milk yield and milk fat is the first two, or protein is the first three top, but there are a lot of also dairy cattle phenotyping, and they are applying this genomic selection not only on the milk yield, so they are applying this genomic selection in many traits mentioned here. Yeah, so that's why very good example of how this genomic selection can be utilized in one herd. And so what is their genetic gain in 10 years, I mean that, so, or 15 years. So they are describing here the very well. Yeah, so you can read this article. And I would like to show you also another another example. This is done by myself, and also with our group did in Washington State University. This is the maybe you know about scrapie. So since you are using the goat and sheep meat, or maybe the cattle meat, you know about the these prion diseases. So these prion diseases are very tough diseases, because when you say, firstly, the students are not very how can I say, they know about the prion diseases, because when you say mention about disease, unless you are veterinarian, so the students are mostly mentioning about the bacteria or virus or the fungal diseases, or like nematodes, means that, so this kind of parasites. But these prion diseases are very tough diseases that's very hard to detect actually. So you can detect these diseases so after you slaughter the animal with brain pathology or so on. Yeah, so in like, for example, getting blood and doing ELISA or doing a microscope scan, that you cannot identify these diseases. So that's why we in the United States, we try to develop a genomic selection for prion diseases, especially for goats. And we understand that, so the prion gene, since the prion gene is very active in prion diseases, so prion gene S146 and K222 positions, I mean that amino acid positions are very active, and in prion disease, I mean that resistancy, and we show that. So this S146 and K222 is when in heterozygous form, the animals are very resistant, and they are not showing any kind of prion disease scrapie in terms of goat. So they are not showing scrapie disease when they are carrying these mutations in heterozygous forms. Yeah, so, and after we showed this one, the United States Department of Agriculture suggest our findings to the all goat breeders, that they say that please test your animals with small number of DNA. So get from, for example, so hair, or from, for example, blood. So test this your animals, and if your animals are not carrying these mutations, we are going to eliminate that animals from the US goat flock. Yeah, so they say that, so this is called this eradication project. Yeah, so if the animals are not carrying these mutations, and if you test, the government says that United States Department of Agriculture says to the goat breeders, we are going to buy your animal and we will, how can I say, kill that animal, and we are trying to eradicate the scrapie disease from the United States goat population. This is also a very good example of genomic selection. As I said, I mentioned to you the genomic selection in a broad aspect. I mean that starting from DNA to the metabolome, but this is luckily, so what this is found that prion protein in goats and in two amino acid substitution is affecting the animals, whether it is susceptible or whether they are very prone to the diseases. Yeah. This is also, I suggest you to read this article that you can understand about how this genomic information, genomic selection could be utilized to select animals against one disease. There are some also other examples for that one in sheep and also in cattle, but this is the very well so established one actually, because the animals was almost so 10 years old when the experiment was, how can I say, done about that one. Since so the animals are infected with prion but still alive although they are in 10 years. Yeah, so it means that it is showing that they are very resistant to the prion diseases. So this is all I can, how can I say, give you information. I would like to thank you all for participating and listening my, how can I say, talk. And if you have any questions, I would like to welcome your questions and we can discuss together.
Okay, Prof. Mehmet, thank you very much. Maybe before we start the discussion, excuse me, could you please stop share from your...
Yeah, sure. I'm sorry. So...
maybe so for someone, for the participant that maybe just join me, we will share about your CV. Yeah, our committee already tried to looking your CV. I'm not sure is it correct or not.
Yeah,
so,
this is the correct one, exactly.
Yeah.
So and you can always contact with me, and so we can develop also joint projects with Turkey and between Turkey and Indonesia, since we are, so as I said, also we need also collaborations in terms of projects, or maybe we can also sign memorandum of understandings or bilateral agreements. So Turkey has some programs supporting the how can I say, bilateral exchange, and also we are in the Erasmus zone, and so Erasmus is also not only making exchange between European countries, they are allowing also to make exchange with the, so I mean that non-European countries. There are some projects making this kind of projects in the Erasmus context. So we are expecting your, how can I say, collaboration calls in other aspects, in student exchange or project funding applications, grant applications, any kind of, we are very open that one. And we have always, so we are already in faculty, agriculture faculty, couple of Indonesian students, but they are making their bachelor in Turkey with Turkish scholarships. They applied to Turkish scholarships from Indonesia and they got the scholarships. As far as I know, we have one student from field crops. She's studying field crop in the Faculty of Agriculture of RGS University. And that's why I would like to welcome you to make a collaboration as well.
Okay, thank you very much. So the committee, is it all? Oh, this is the education. Okay, so, okay. This is your paper. All of the paper, yeah, okay. Next, maybe. Oh, very good. 17, HI index, right? Okay, next. Okay, next. That's all, the committee? Okay. Okay. Oh, okay. So, I believe we have maybe one from the participant, or no one right in the chat room. So maybe anyone would like to ask for Prof. Mehmet, please raise your hand or maybe just open your mic so we can hear your voice. Anyone? Okay, Mr. Murshid. Okay, go ahead.
Good morning, sir.
Yup.
It's a nice presentation, Professor Sir, and I have learned a lot of things. It's a tough topic regarding genetics, genome sequencing. It's a tough topic subject, I think and believe, as because I am a animal husbandry graduate and MS in dairy science from Bangladesh Agricultural University. We have a subject genetics and breeding. So our honorable guest professor sir, he has presented his topics from the research background, and I have learned many things. Heritability, pedigree selection, sire and dam selection, QTL, and many things, and pharmaceuticals industry background, and another workflow. That is the very important things, I think, and another genotypic things, and most postmortem muscle is also a very interesting things, and color and pH level. We want to know. We can determine the muscle quality on the basis of pH level. pH level below, we know that below 7 is an acidic condition. This is a question when the pH level is more than 7, how can reduce it? This is one question, and another question, just to curiosity to know. Another question is that dairy cattle. Our honorable professor has been present in his topic, dairy cattle trait, that is milk production, muscle production. There are so many genes, at least 13 genes, so far I learned, and 222 amino acids. We know that the 24 amino acids, essential amino acids, valine, isolin, lysine. In the dairy cattle, we know that methionine and lysine that build our brain cell development, and in that case, we have to suggest every person to drink milk, I think. There is another question is, how can we accelerate our heterogeneous show in the dairy cattle?
Okay.
Yeah.
Okay.
Can I able to understand the question?
Yeah. Okay.
Excuse me, sir. Thank you very much.
Okay. Thank you, Murshid. Okay, Prof. Mehmet, have you?
Yeah, I heard. Yeah, so yeah, I heard. Maybe so. I try to answer one by one, and then maybe we can also discuss together.
Okay.
Mr. Murshid, thank you very much. So we have a lot of interaction with BAU actually. So Munir Hussein, maybe your colleague. Yeah. So Munir Hussein. So he was my former colleague. Yeah. So from Bangladesh Agricultural University. I think you are from BAU. So Bangladesh, or you are from Bangladesh? Yeah?
Bangladesh. Yes, Bangladesh.
Bangladesh. So I have a lot of colleagues working there actually. So Munir Hussein.
But I have servicing in government service, another academy, Bangabandhu Academy.
Okay. Maybe.
Yeah,
so, but anyhow, so very pleased to meet you. The first question is, so how we can decrease the pH? So there are, as far as I know, the first way is actually, I mean that if you would like to make a desired pH, so the one of the method is genomic selection. So if you identified your population, so the variation in your population and which way you would like to go on. So for example, through the basic level or through the acidic level, you can decrease or you can increase your pH level by genomic selection. But for another method, as far as I know, so there was one colleague in our department, for example, he gave a sugar solution to the meat type of animals. So he tried to understand the sugar content of the muscles, and as you know, the sugar content, the glycogen content actually is the main determinant of the pH. Yeah, so of post-mortem, I mean that. So, but this is not a very practical method. So because the sugar is not very cheap, I mean that. So it's not. But as I said, so this was done because the nutrition is affecting the glycogen content of the muscle. Yeah, so and the glycogen content is affecting the post-mortem pH. This could be changed by this kind of methods, but as far as I saw, as long as I said so, these are not cheap methods. The best way is identifying the variation among your population and to select animals which way you would like to go on, to acidic level or to the basic level. Yeah, so the genetic methods is the not I say that cheapest, but the most reliable, most stable one, because nutritionist things like feeding the animals with, for example, a high glycogen content of feeds or giving sugar water or whatever, it may affect or not affect. So, but there are several methods as I know. So I don't know whether it could be the right answer. The second question, that's I cannot understand very well actually. So could you please repeat the second question?
Okay, Mr. Murshid, please repeat.
Yes, sir. I just to know about the to clarify the heterogeneous amino acid, how to increase the quality of muscle or milk production. Just to clarify, nothing else.
Heterogeneous amino acid or
in your paper you have
you are asking about I'm sorry, my internet connection is not that much stable. Yeah, so maybe could you please write in the chat section that
yes.
That could be more, how can I say, I can understand maybe.
Next time I would contact you email.
Okay,
email. Yeah, thank you very much.
Sure, that's yes. The best way of actually.
Yeah,
thank you very much.
Thank you very much, sir.
Okay, thank you very much, Mr. Mohammed, and also Prof. Mohammed. Maybe we still have maybe one more question, if there is some question, short question from the participant before we are no one. Okay. Okay, and then because of no one, so as a closing remarks maybe from your lecture, Professor Mohammed, maybe this is a very good, your lecture is a very comprehensive presentation about meat quality from the genetic point of view. Yeah, it's very complete, I think. Yeah. So from our department, from the Committee of Summer Course, we would like to give you a certificate for for giving the lecture. So I invite Dr. Jakaria, our Secretary Department. Dr. Jakaria.
Okay, I'm here. Thank you.
Okay.
Thank you, Dr. Sri Darista.
Okay.
Thank you, Prof. Mehmet. Yeah, on behalf on the Department of Animal Production and Technology.
Thank you very much.
Faculty of Animal Science, Bogor Agricultural University. I would like to deliver a certificate of appreciation for your nice lecture and your support of our online summer course program today. Thank you very much, Prof. We hope to collaborate again in the future. Okay, thank you.
Very much.
Okay, thank you very much, Dr. Jakaria, for giving the certificate for Professor Mehmet. And before we closing your session, then one of your colleagues, Professor Asep, would like to have some chat with you. So please go ahead.
Halo, Profesor Ulas.
How are you? Say
Yeah.
thank you very
So
much.
actually, we would like to
Eid Mubarak.
thanks to you. Otherwise, you are in vacation, but you accept our invitation. So it is, we really highly appreciate to you. And then, yeah, normally vacation we cannot disturb anyone, but
No
in your
problem.
case, you
So with
accept
pleasure,
our
with
invitation.
pleasure.
But
So
yeah, we are really thanks, and we hope we can continue for the collaboration, like you mentioned, maybe either in education purpose and also maybe in research based. Since here we have a lot of participants, we have 33 university Indonesia, and also we have 20 university abroad. So we hope by this lecture or later on, maybe we can develop the next collaboration. Thank you a lot, and greeting also from us, from here, from Indonesia, to your country and to your university. Thanks a lot.
thank you very much. Thank you very much all. I will hope to see you again. Yeah. So in future. Thank you very much.
Very much, Prof. Mehmet. Okay. Okay.
Have a good day.
Thank you, Prof.
Eid
Fermi.
Mubarak.
Jazakumullah khair. Barakallah.
Okay. Thank you. Okay. Now we'll continue our program with the overview of Indonesia culture from Bali. Local wisdom of Indonesian animal, the Bali pig. Please, committee, if you want to share the video, maybe.
A mesmerizing getaway. A holy sanctuary. A captivating culture. Sublimely sacred. Yet also excitingly profane. The ultimate paradise. A luxurious escapade. Here you have some of the hippest spots on the planet. Release your inhibitions for an adventure of a lifetime. Whether you fancy the beach or golf, just love food served exotically. Or adrenaline rushes. Enjoy oases of tranquility. Where pampering is art perfected. Mysteriously festive. tourism with its own history. Branded hospitality. The ultimate travel destination and more.
Okay. I believe the video is over. So for the next session, the afternoon session, the second session, if anyone would like to have some asar prayer, just leave the summer course, maybe maximal about maybe 10 minutes. Okay. So after that, join back again to the summer course again. Okay. I think that's all for the announcement for now. So go back again to the committee. Go ahead. And we're still waiting for Dr. Vincent, right? Is Dr. Vincent already here?
Hai pemain 1, siap hadapi
Oh, how are you, Dr. Vincent Guyol?
Okay, you sir.
Okay. So, do you like to start now or maybe some after 3 p.m. or sharp at 3 p.m.?
It's up to you. You're the boss.
Okay, it's up to me. Okay, so maybe your session will start at 3 p.m. But maybe now, please committee, can you show me the Dr. Vincent CV? Committee. Okay. Okay, this is Dr. Vincent. Am I addressed right? Guyonet or just Guyon? Is it?
Guyonet, yeah. Just say Vincent, Dr. Vincent. That's fine.
Okay, Dr. Vincent. Okay. Next. Sorry. Back again. So right now, Dr. Vincent is managing director of FFI Consulting Limited at Brockville, Ontario, Canada. Is it right, Dr. Vincent?
Yes, it is.
Okay. Then next, please. So an education, doctor in veterinary medicine at Lyon, France, and also the PhD of poultry science and from the University of Georgia at the year 1991.
Yeah, some time ago now.
Okay. Okay, next. So this is your, what I call it?
Okay.
Yeah.
Where do you get that from?
I'm not sure. As the committee.
Okay, okay, no, that's fine. But I mean, you don't spend much time on that. Don't worry about it.
Okay, okay, okay. So you are now the consultant to the poultry sector. Okay, from 20.
That's what I'm. Yeah, don't worry about the rest because
Okay.
I wasn't sure if you were going to introduce me or how much detail you have. So my first slide for the students, I was just explaining my background, but
Okay, okay.
I won't have to do it. But don't worry about it.
Okay. Next, please. Okay, you are also author of the 4 book. Okay, guest lecture at Jilin University and University, China. Next.
There you go. Wow. So, yeah, I don't know what. Okay, what's that? Oh, don't worry. Yeah, okay.
This is the Google Scholar for you. Okay.
Oh, goody.
Yeah, you have a HI index around 6 or 4. I'm not sure. Okay. Oh, okay, okay.
All right, I like that one.
So.
Oh, look. I don't know if you can see.
Oh, from Indonesia, right? No?
Of course.
Of course, yeah.
Of course. I've been, here we go, put it there. I've been, we don't see it. I need to find somewhere where you can see it. Let's see.
I think the first place is the better one.
Yeah, same middle. Anyway, that's up.
Yeah.
All right.
That's perfect. Whip.
Yeah. Yeah, I've been to Indonesia about 4 or 5 times, I think.
Okay.
Last time was in 2017.
Okay, how do you think about Indonesia at that time? General
It's I love it.
You love it?
I love it. It's nice. I mean, just like Southeast Asia, everything is growing. There's so many opportunities, And in the livestock sector, the poultry sector, this is fantastic field. If you look at Europe, there's more and more regulation. There's more and more issues, right? So production in Europe is really declining because of so many things. Whereas if you look at Asia, there's so many opportunities. There's so many, expansion of the business, interesting things. So yeah, it's very good. So
okay, without further ado, so the time is yours, Dr. Vincent. Could you do the share screen, or our committee do the share screen for you?
Yeah, no, I think I'm gonna try that. Is the share screen? Share. I need to do this.
Okay, already see it.
And then I need to duplicate the slide. Okay, is it okay now?
Yes, it is okay. Right.
All right.
Time is yours.
Okay, so you give
maybe before 20 minutes before 5 p.m. around Indonesian time. Okay.
Right. Yeah. So are we live? Everything is good with the students?
Yeah, everything live with the student. Yeah, we are live.
Okay. All right. So anyway, so we went through that introduction, so I won't have to talk more about it. So the topic for my presentation is really to look at the impact of the COVID-19 that will have on the poultry and egg sector. And you will see that, so first, I will look at some of the key observations that we made, things that we learned during the COVID pandemic. And then from there, I will present 6 of the key, what I look at, key trends for the post-COVID-19 world. And you have seen them here listed. So we're talking about growth of the sector, development of artificial intelligence driven technologies, branding, marketing of products, potential, the new frontier, the further processing of both eggs and meat, appealing to new consumers and consumer needs, and finally communication. So first check on the sound and am I speaking fast enough, too fast? Is it okay?
I think okay.
All right.
Okay.
Okay. So let's have only one brief slide for COVID-19 and to show you what we've learned in a number of countries. So here we are. Key learning really for the sector, although they can probably apply to more than just the poultry sector. You know, one of the key issues right at the beginning of COVID was really ensuring the safety of farm workers and also making sure they could come on farms. You know, we struggled. A lot of companies struggles. There was restriction in transportation, movement of people. So really having people on the farm doing the job and making sure that they were trained for the job that they were supposed to do was critical. Another one was because of, and it's true for both the egg sector and the poultry sector. So when I say poultry, it's chicken meat, okay? Shortage of packaging, because typically product that you sell in retail stores, grocery stores, are very different from the product you sell in restaurants. If you look at the birds, for example, the birds that you buy in the store are typically smaller than the birds that are used for further processing for KFC, for example. So different type of products and different type of packaging. So we had a problem of enough packaging for the type of products consumers wanted at that time. And it goes with that other point was the rapid shift in market needs. Again, the food service or the restaurants were closed in many countries. So a whole segment of the sector, the products were not being sold, could not be sold through restaurants. And it was hard to, as I mentioned a minute ago, say with the size of the birds, to sell them into retail stores. Consumers' behavior also have changed. I mean, we saw, I don't know, but in Indonesia, likely just like other countries, but suddenly a lot of people have been ordering food online and they had to cook more at home. In North America, a lot of people, when they have any breakfast, they would just drive their car and pick up in a fast food restaurant and go to work. So really changing behaviors, changing consumer preference. And finally, you know, the last point is all about communication, that we realize that it is critical to communicate to consumers, to customers, and also to our employees to make them feel safe, so that they are comfortable coming to work. And because, as you know, when you manage animals, you have to have workers on the farm every day. Animals, require our attention every day. So that's a very quick overview, but that's sort of the key learning. And from this key learning, we can go to some of the different trends that we can expect in the post-COVID-19 world, which I hope will start very soon, starting in North America and Europe and other countries, depending on how quickly you can vaccinate the population. We will all be there. So if we look at global animal production, so that's terrestrial animals, so no fish here, no agriculture, you can see that between 2010 2019, chicken meat grew by 35% and the egg grew by 30%. So tremendous growth and at much faster pace than milk, sheep, cattle. Here you can see pig meat grew for that period only 1.2%. That's the impact of African swine fever. You know, in the past, you would have seen pig meat probably growing between 15 to 20%, but this is what happened when you have African swine fever across almost the world, at least certainly in Asia. If you look at the 30% growth for eggs, those 19.2 million metric ton additional, it represents about 42 eggs per person on the planet, additional eggs, all right? So that was about 330 billion eggs. So a lot of additional nutrition delivered through increased production. Now, if you look at the countries producing, you can see here the top 10 lists, and very happy, and you probably know that Indonesia here is ranked as number 4 with the 4.7 million metric ton. 5.7% of the world egg production is in Indonesia. You see China is the big giant in egg production, over 28 million. US is about more than 4 times less, India and in Indonesia. Overall, top 10 countries, almost 71% the world production, and you can see that we have quite a number of Asian countries in this top 10 list, which is reflected by this other slide. You can see globally, 2019, 83.5 million metric ton were produced for eggs, and 61% were coming from Asia. Europe used to be the dominant player at 12.9%, now second. Latin America growing at 12%, and North America 8.7%. Same type of slide. Now we're looking at chicken meat production. So all my slides, when I will say egg, it's typically, it is chicken egg, okay, and not other species, even though I know that, and I have a few slides where I talk about the fact that you also have a high consumption, especially in Asia, of quail eggs and duck eggs, for example. But here all the data is chicken egg. And for chicken meat, we're talking about the sort of the modern more intensive, the broiler type of chickens, okay, not the native bird. So again, here, Indonesia ranked as number 6, 3% of the global production of broiler meat. And you can see that compared to the egg, less concentration. So because the top 10 countries represent about 60% of the world production of chicken meat. And again, here, Asia, 36.3%. Again, a larger production in Asia, but Europe here has a bigger share, 16.5%, and Latin America certainly with a country like Brazil at 22%. So Southeast Asia, so I have listed here 8 countries on the right-hand side, and you see Indonesia here is second. They're all listed by with the flag and alphabetic order. So you see the growth over the past 10 years. Chicken meat around the world grew at 35%, and in Southeast Asia grew by 76%. The chicken egg grew 30% globally and 132% in Southeast Asia. And other eggs, so quail and duck, grew 23% in the region compared to 14% around the world. So really, Southeast Asia, including Indonesia here, is really growing at a much, much faster pace than the rest of the world. The 132% for chicken egg, you may guess one of the reasons that huge jump, it's really what happened in Indonesia. And you probably are aware of, so this is here on this slide, egg production and evolution of the population in Indonesia. So population is in blue, egg production. And in 2017, they assumed it's the government, at least the statistic reported for Indonesia, had a huge jump in production. We probably a new way of reassessing production in Indonesia. So it's basically, I won't say overnight, but the data from one year to the next imply that basically there was about 160 million more laying hens producing eggs. So that was a huge jump in those numbers, which means also that in terms of consumption, we went potentially from, although I mean it's not the way it is, but from 95 eggs to 293 eggs consumed in Indonesia. So the point here is that whenever we talk about animal production, statistics or numbers, values, market data is extremely important. Imagine if you're trying to sell eggs, if currently the consumption is 95 eggs per person, it's a different approach than if the population consumed 293 eggs. So Anyway, those are the numbers, and that's sort of why we have that huge increase in production in the region, mostly driven, at least part of it, with that revision of the numbers for Indonesia. If we look at now the meat consumption in Indonesia, again, so the broiler, for the first table here, you can see that based on the OECD database, Indonesia, there's a consumption of 7.9 kilos per person per year. Compare that to the leader in the region, Malaysia, 49.3, Vietnam, 16.5. The global average is 14.9, so Malaysia is about 7 kilos below the world average, if you wish. And the world champion in terms of chicken meat consumption is Israel with 64.4 kilos. If you look at other type of meat, and I put only here beef, again in Indonesia, you see 2.2 kilos compared to the world average of 6.4. So slightly lower for Indonesia compared to some of the neighbors, Malaysia and Vietnam. And again, the world leader in terms of meat consumption, beef consumption, is Argentina with that 36.9 kilos. So in terms of consumption and global growth, what are the key factors that help grow the food demand? It's really some demographics, so population growth, economics, the income growth. So those are managed by, at macro level by government and so forth. But the next two elements, the trends in dietary patterns, the consumer preference, the sector through marketing, through advertising, through promotion, is able to modify and convince consumers to eat more eggs and more chicken meat compared to eating fish or eating beef. So those are the key elements. So in terms of the top two drivers, population growth and income growth, well, we're very lucky and fortunate in Indonesia because those are really, good numbers in terms of growth. So this is a very, very recent publication from the FAO and OECD. That's a yearly document, Agricultural Outlook, and I think I have the reference in the previous slide. But you should, if you're interested in sort of forecasts in production and what's happening in consumption around the world, this is an excellent document. Except that it does not cover at all eggs, but it covers every other main agricultural commodity, crops, but also meat, fish, dairy, and so forth. So it's quite interesting. So you see prediction by 2030, population would increase by 800 million, and India would become the most populous country. Income, again, as I mentioned a minute ago, income level is important in terms of food consumption. So globally growing at 1.9% per year. But you can see that Indonesia is going to grow basically that's twice that level, so 3.8%. So again, more opportunities through increased income available for food to purchase higher value animal protein, and hopefully, it could be chicken and eggs. Poultry production will increase again about 22.8 million ton. And the poultry production will represent the growth. 52% of that global growth in meat production will come from chickens. Okay, so really a clear emphasis of the role of poultry meat in the growth of the whole sector of poultry production and also consumption. As you can see in the next point here, 51.6% of the total growth in meat consumption will be coming from chicken meat. In terms of the economics and the recovery, see at the world level here on the left, economy decreased by 4.4% and increased hopefully in 2021 by 5.2%. So nice recovery, which help obviously having people spend more money and consume more animal protein. And if you look on the right-hand side of the slide, emerging and developing Asia, so expectation is 8% growth this year, meaning again, so that, and once again, Indonesia should be able to recover at a much faster rate and pace than the rest of the world. So those, again, FAO, OECD data, we can look at their prediction in terms of chicken meat consumption. And you can see that for Indonesia, it is expected by 2029 to grow in Indonesia by 17.7%. If you compare on the global basis, growth only for 1.3%, but 17% in Indonesia, faster growth than Thailand, slightly less than the Philippines, but really, again, phenomenal growth expected for the sector. If we look at egg consumption, so on the world, based on global production and the population of the world, the average is 180 eggs. In Asia, it's 187. If we look at Southeast Asia, it's 198. But within 198, you have countries like Malaysia, 368, Indonesia, 293. So really, if you take those two huge egg-consuming countries, the rest of Southeast Asia is 109, which will mean also that there is opportunities to increase consumption in those countries, and that we'll see later on maybe an opportunity for Indonesia to start exporting eggs to some of those countries. So again, growth opportunities. You can see here the summary of the so on the left, that's the global world average, 180 eggs and 14.9 kilos of chicken meat. The center is Indonesia, so 293 and 7.9 kilos. And on the right-hand side, those are the top world leader in terms of consumption. So you see the country Mexico, 372 eggs per person per year. And for chicken meat, it's 64.4 kilos of chicken meat consumed by every citizen in Israel. So that was my first trend in terms of the global growth. Second trend that I would like to talk about, it's the use, and here AI is not avian influenza, but it's artificial intelligence, artificial intelligence-driven technologies. And I think through the COVID-19 pandemic, a lot of people have had to experience with new ways of working, teleworking, video conferencing, webinars, just like this event that we have now with me all the way from Canada talking to you. So, and COVID-19 has sort of forced us and forced the sector to reconsider how they work and how they adapt to new technology. So I think overall, the COVID-19 is going to help us accelerate the use of those AI-driven technologies. So artificial intelligence, sort of very briefly, so some slide to introduce the topic. Basically the goal is to create a machine that can replicate the human brain. The human brain, when you think about it, it's a fantastic structure. You can see that we have about 85 billion neurons, 10 to the 15 synapses, 180,000 kilometers of wires of connection between neurons through the synapse, and also very low energy consumption of the brain. And also the most amazing part of the human brain, I guess, is that learning is really part of our intelligence. So we are, when we're born at the beginning, we don't know things, but we have the capacity of learning, and sometimes learning by ourselves, learning new things. So really, some people say that artificial intelligence, it's basically trying to find a machine that can mimic, replicate a child's brain, because within that child's brain you have the ability to learn. It's not downloading all the information that is available, but it's creating a machine that is able to think and learn. Okay, so that's the ultimate goal. We're not there yet, but people are working hard on it. So one of the challenge in the poultry sector, and I think in a lot of livestock animal production sectors is that people capture a lot of information, but it's mostly done the way we can see it on the picture on the right, by hand, writing, scribbling, and it's not so much yet sort of the more fancy way that you can see here on the picture on the left, with a nice, sort of computer generated printout of your data and so forth. And the key challenge for the livestock sector, but it's not just the livestock sector, it's all industry sector really, is that we have a lot of data, but 90% of the data remain untapped because it's not digitalized. It's not available to us easily to analyze and to interpret the data, okay? So 10%, we have it on computers and we can do something with it. 90%, it's extremely hard. So that's the challenge. And so here's an example for the egg sector, but poultry, the meat, chicken meat would be the same. We collect a lot of information. So production parameters, the bird weight, the feed consumption, mortality, egg production, egg quality, all those different things about the birds, maybe some data on serology, so how well the chickens have been responding to vaccination, and you test also the environment for the presence of salmonella. So we have a lot of data, and most of them, as I mentioned, are collected by hand, people writing down. So the trend is that we are moving more towards what I call, connected farms. Basically, the measuring and the recording of the data is done electronically. So you have sensors, you have Internet of Things that collect the data. So say temperature, moisture level, humidity in the barn, moisture level of the chicken litter, for example. This all could be captured automatically for us. Then the data is transferred. You can monitor the data, and then really the last step, the final step, this is the sort of the true benefit of artificial intelligence and all those connected devices, is that you can analyze the data to make predictions, okay? So there is some estimates that by 2050, the farms will generate 4.1 million data points through those Internet of Things, sensors, devices. So which means that with that huge amount of data, it's going to be impossible for the human brain to be able to interpret the data. We're going to have so much data that we are no longer able through our own thinking process to understand what the data is saying, okay? And that's when really we need artificial intelligence to help us. So another example, or just to make the point, let's look at, again, egg production. And we can think of what are the parameters that can affect the production of eggs in chickens. So you can say, well, there's the age of the chicken. There's also the age of the bird when she starts producing eggs. There's the weight of that bird when she starts producing eggs, the level of uniformity of the flock when they start producing eggs, the feed consumption, the water consumption, the protein level in the ration, all those different things. You see a lot of factors. And again, think of human brain. I mean, equations, we can deal with 1, 2 parameters. When you start having 10, 15, I mean, this is impossible, right? And there's a lot of combination and everything. So what's the best formula? We don't know. We don't know by ourselves, and that's where we need those machines. So machine learning or artificial intelligence, machine learning, you have 2 types broadly, okay? I will go briefly on that. What we call supervised learning and unsupervised learning. Supervised learning, it's basically when you have a lot of data where, from my previous slide, for example, I know the weight of the chickens, I know the temperature, I know all those different parameters, and I know the outcome. I know that under these circumstances, the chickens were producing so many eggs. So I can train the machine, and I can tell the machine, if you see these parameters, these values, this is the type of production you can expect. And by having a lot, millions of data points like that, you can train the machine, and the machine then develop a system that it knows, if I see all those different parameters, this is my best evaluation of the production is going to be that way. So that's supervised training, when you have a set of training data points that you can train the machine with, and after that the machine presented with new numbers is going to estimate the outcome. And then you have the unsupervised learning, which is even more, interesting, or do the other type of things, is that basically here you just feed the machine numbers. You have no idea the outcome and so forth. You just ask the machine to look at those numbers and see if it can see any trends. That's what you do, for example, when you do what they call clustering or market segmentation. So let's say you have customers and you have 1 million customers in your database. You have all those numbers about the shopping preferences, where they live, all those things. You give that to the machine, and the machine is going to find clusters. It's going to find groups of individuals that have sort of similar pattern, which then after that, potentially you are able to target more on the marketing side, because they have common characteristics that are easier for you to target. So, sorry. So you probably, or you may have, heard about those neural network structure. So that's basically the machine that is trying to mimic the human brain. Can you remember the human brain? You have neuron and you have those synapses. So the neurons here would be those round structures, and the synapses and would be those connections here. So deep learning. So you have layer 1. That's the input. That's the different parameters that you enter into the machine. Then you got different layers, and layer 4, and then you would have the outcome, the results. So deep learning means that you have more than one layer. Okay, so if you hear about deep learning network and deep learning machine learning, that's what it is. So again, how does it work? Again, as I mentioned, if you look at on the left of the slide here, you have training data. So all my different parameters, which in artificial intelligence jargon, they call that features. So you got those different features, those different x1, x2, x3 that that and you also know the results, the final weight of the bird, which is y. So you feed the machine all those x numbers and the y's, and the machine, through repeated process, is going to give you develop the machine learning algorithm, and then in the end, it's going to be able to predict you the final weight of the bird. Okay, and that's a system that goes extremely fast, repeatedly, and so you train the machine with some of the data you have, which means that first we need the data, and after that you can make prediction. And that's really the beauty of artificial intelligence, is that through that vast amount of data, the training and so forth, you are able to make predictions into something, an outcome that you either want or do not want. And therefore, if the system predicts that something's going to happen that you don't want, then you have enough time to correct it so that the results is different. So let me just explain in my next slide here. This is from a company. Again, I will present through my slide a number of commercial applications. I just want to tell you that I don't have any financial interest with any of those companies. I just use them to illustrate a point, okay? So here, this company, Intelia in Canada, is getting through Internet of Things and sensors a lot of information from chicken houses during production, right? And with those numbers, they have developed an algorithm where they can basically tell a producer 14 days in advance what will be the weight of the bird, and with excellent accuracy. And obviously, the more data, the more accurate the prediction is. So let's say you are a broiler producer, and you need to produce a chicken, say 2 kilos, 37 days, for example, and you send it to a processing plant. So, the processing plant, they want always to have a bird that is always the same size, because for the equipment, it's easy, right? If all the birds are the same size, it's very simple. If you got big birds, small birds, it starts to be a problem for them to manage the plant. So they want the same size of bird. So you're the farmer, and let's say 10 days before you need to send the birds, you get the prediction and tell you that the birds are going to be slightly under the target weight. First, in some cases, the farmer will also be penalized if the birds are below the target weight. Okay, so you want to avoid that, and you want to have birds uniform and so forth. So 10 days in advance, your birds are going to be slightly underweight if you don't do anything. So what you can then do, you can either modify you have 10 days to stimulate activities of the birds to increase feed consumption so that they grow faster, they grow more, and they reach the target weight. So more lighting, nutrition, and so forth. On the opposite side, if the prediction 10 days in advance tell you that your birds are going to be too heavy, again, a penalty for that. Yes, I saw somebody raise hand. I finished my sentence, and then you can ask the question. Then if the birds are overweight, then you can also slow down the growth and so forth. So yes.
Okay, who is raised hand, please?
Oh, maybe what I can do, I don't know, if nothing, I've got those 6 trains. I can stop at each train, and we can see if students have questions, or they can send them to you. I don't mind. We can stop whenever there's a question too. It's whatever works for you.
Okay, actually, we would like you to complete all the presentation first, and we will have the discussion session after that.
Okay.
So if someone that raised hand, maybe he or she can write down at the chat room. So after we have completed your presentation, then I will go reading their about their question. Okay.
Okay.
Okay. So please continue, Dr. Vincent.
I will continue. Yes. And actually, so I'm trying to illustrate a bit more some of those latest applications. So here we are going to look at a short video of robots. So we have also, the new trend is using those machines inside the barn. So let's have a look.
Excuse me, Dr. Vincent, is it the video come out with the audio or just?
No, it's just to show the birds.
Okay.
Yeah. And maybe if I can speak at the same time, but this is basically, breeder birds, so they lay the eggs, and the machine was designed to really motivate the birds to go up and lay the egg into the nest box, okay? And because that's a challenge that you have to face. If you have the eggs lay on the floor, they become dirty. If it's a breeder flock, then you have a risk of potential, contamination and low hatchability. So you want always the birds to go and lay the eggs in a nest box. And it's a lot of work to walk through the barn and move the birds up. So here they use those little machines to convince the birds, to motivate them, to entice them to go to the next box. Okay, sorry. So that's basically one application. Another application of those new technologies will be also for broiler chickens. And some of the advantages that you see is that the robot is present on the farm all the time. So works 7 days a week, 24 hours. And farmers have been noticing that with those machines, you have increased weight gain and also reduced feed conversion. So the production parameters are also excellent, and the robot will motivate the birds, to move around, to go to the feeder, so they can grow faster. And one other element is also, and let's look at I have another video to show you, and then I can explain sort of the benefits of what of the value of that. And I think there's no text here. So broiler chicken, broiler house here. Again, you can see a lot of birds will be down. So here's one of the elements that is very interesting. The baking is going to also help to turn around the litter, so there's no water accumulation, there's no wet litter, which reduce the problem of food burn, food pododermatitis, and so forth, breast burn, and therefore increase the welfare of the bird. So that's true benefit that brings both value in terms of productivity, but also value in terms of welfare. Again, two or three benefits here. The machine motivates the bird to move around. If they move around, they're going to eat. And also it turns around the litter all the time. So you have space, water, moisture, and wet spots. Self-driven, all automated, sort of one of those applications again of artificial intelligence here used. All right, and then you also, so there's a number of robots now that are being available, and this one here is also at the same time combining artificial vision, and you have heat sensors, and it collects a lot of information. And I'm going to show you here on this next slide. For example, they can collect, so the robot goes through the farm continuously, and you can have information about ammonia level, that's the top, about the temperature, that's the middle one, and then humidity level. And with that information, basically the farmer can easily see if within the chicken house there is somewhere a problem. Let's say you find a spot where there's high humidity, maybe there's a water leakage, or you can find temperature fluctuation. It could be also that there's a problem of ventilation or something that, so really it's a nice tool that it makes it much easier for the farmers to detect where they can potentially, have a problem. And other type of technology that we see and probably adopt is, the use of drones to monitor when you have chickens going outside, what we call free range chickens, or organic chickens also would be the case. So you can use drones to monitor the birds, to prevent also the presence of wild birds that can either, attack our chickens or also transmit other type of, diseases. So overall, the value of artificial intelligence technologies on the farm, it's really all about providing, using current field data to help you make prediction, and those prediction on which you can then take action, and therefore you can hopefully modify the outcome if the outcome was going to be negative for your production. Artificial intelligence also is through robots and other element that we can have on farms is going to reduce tedious, hard manual labor that is in many countries harder and harder to get because there's not enough people that are interested in working in farming. Advantages in productivity, more profits. So again, it's good for the farmers. They invest in some equipment that will deliver more return for them. And then finally, and it's a very important one in a number of countries, Western countries, where there's more and more emphasis on animal welfare, bird well-being, and so forth. And those machines are able to also help farmers. They are continuously 24 hours a day, 7 days a week present on the farm, and they do facilitate the work of the farmer to monitor the well-being of the birds. Okay. So just sort of the key 8 elements of artificial intelligence technologies that you can see listed here. And we looked at here mostly through those machine learning that helps you do the planning and control. And I will show you later on some examples about application of visions and also some work being done on natural language processing, here being the language of the bird. So there's some system trying to interpret sounds made by the bird to analyze what is happening on the farm. So the next application for the sector is also using artificial intelligence technologies to help in a processing plant, where you have a lot of painful, sort of repetitive tasks, a lot of manual labor. Also, the problem is that it's very hard for managers you don't have real-time data, so people process chickens and maybe only at the end of the day or at the end of the week, you can look at the numbers and say, oh, the yield, so the amount of meat we got from the chicken carcass was down from last week, or we did not do a best job in terms of getting as much meat as we can from the chicken carcass, for example. And then obviously, if you don't have that real-time data and by workers, it's hard to give incentives, so pay premium for good workers compared to workers that do not do a great job. And then finally, one of the issues that when you do the processing manually, the speed of human intervention is much reduced compared to what machine can do. So we see, is an example, very simple robot being used for egg in the egg business, just to carry those eggs. Machine, very simple, but it helps because it's very labor intensive for workers that you have to do that for 10, 12 hours in a day. The machine does it, no problem. You have for processing, meat processing, again, that was the challenge I explained a minute ago, where let's say chicken breast, which is a highly valued premium meat in Western countries, we typically eat the chicken breast and that's it. So processors want to make sure that they get as much meat from the breast as possible, right? And typically in the processing plant, it's very high speed and it's very hard to monitor how well the workers are doing the job. So here is a technology using artificial vision, near infrared, to analyze very quickly the carcasses and how much meat is being left on the bones, right? And that with that information that is instantaneous, then a manager can go back to worker C or ABC. This worker is doing a great job. That other one is not removing enough chicken meat, and therefore maybe it needs to be retrained or re-explain the task and so forth. So really a lot of opportunities to increase productivity and efficiencies in the processing of chickens. You see that also those artificial vision in the egg processing business. So here it's basically, well, you have the shell egg, it's being cracked, it's being opened by those machines, and you're trying to recover either the whole egg or the yolk or the white. And here you got artificial vision that is going to make sure that it's being done properly, because if you want to have egg white, so it's mostly protein, it's got great foaming characteristics, but if you have a little bit of yolk, then it's going to lose all those characteristics. So therefore, if you sell egg white to bakeries, to make cakes, to the food industry, they want to make sure that the egg white does not have any piece of yolk, and this can be sort of monitored through artificial vision. So again, another picture of this machine that you can see on the right-hand side, the computer monitor that is giving you the picture of each of those little cups that recover those eggs, to indicate whether or not there was some presence of the yolk in the egg white. You also have artificial vision being used to inspect the eggs when the table eggs, so people that are going to buy in the grocery store and consume. You can see that one of the advantages in the past, it was done by, visual by humans, but now you can have a much faster speed of processing by using machines that will do that for you. And I will show you here little examples from a grading station, so a plant where eggs are being washed. In some countries, you wash the eggs and then you inspect them and then you package them. So here's a little video. All right. Oh, sorry. And then sort of what the computer sees and tells you. So again, every egg, and you saw the speed at which they were going. I think it's about 70 eggs per second. And the computer is able to track every single egg. And if there's any defect, the computer is going to say this egg has to be moved to this lane, or this is rejected, and so forth. So really amazing development of those technologies to facilitate the speed, the processing efficiencies of processing of food. So here you go, and you see those different lane of the machine. So depending on some, maybe dirty eggs could be coming up front here, and the nicely packaged egg will be in different lanes. Okay. And again, this is just to illustrate through the machine learning process how we go about it, and it's extremely complex. If you think of vision for us, it's something that comes naturally. We don't think about it, but for a machine to see and to interpret what it sees, it's extremely complex, because the machine is looking only at a few pixels, a few little square of a picture, not the whole thing. It's impossible. So through a selected vision of those tiny little spots, the machine is able to interpret and understand that it's looking at an egg that is misshaped, that is an egg that is dirty, that is an egg that has internal defect like the blood spot and meat spot and so forth. So again, great development, and it's mostly that it helps us process those eggs maybe at 2 or 3 times the speed that you would do it if a person has to inspect the egg. And the same, obviously, those type of artificial intelligence driven techniques are being used more and more now in processing plants, meat processing plants. So I'm going to show you now with this example from a company from Japan, a short video. Again, no text, but just look at it and there's some captions of what's happening. So here it's all about packaging and value, high value chicken breast, and making sure that only the products that are perfect are being packaged and the others are being eliminated and would go into other products. So you can see how quickly the machine is able to see where there's a chicken breast, and if the chicken breast is without any defects, so therefore it can go in those plastic packaging right here. And when there's some defect, you can see the machine is going to put them in the back there, in those bins. Of bulk packing. Okay, all right. So basically, you know, again, for in the processing sector, it's all about either getting real-time data so you can quickly react if there is a problem and bring corrective actions. It's also about reducing the amount of very tedious, labor-intensive, repetitive manual labor that you need in a plant. And overall, you know, you can reduce losses, improve productivity, also, you know, the quality of production. You have less defects, you have less consumer complaints. You also have a better, you know, sort of welfare for the workers, because some of the very painful tasks can be done by machine and no longer by them. In the future, some of those other applications that people are working on using artificial intelligence would be, for example, using egg production data. You may be able to predict that within 3 to 5 days, potentially your flock the production is going to drop because of some element. The machine cannot yet tell you why the production is going to drop, but it can tell you that it's going to drop. And then it's up to the farm manager to verify all those parameters and look at it and potentially correct the mistake, right? But basically, so artificial intelligence can make prediction, can tell you within 3, 5, 10 days, this is the result that you will get, but it's not going to tell you, but that's because you have a water leak in this section of the barn, right? You still need people to interpret and to analyze all the reasons, right? But at least artificial intelligence is helping you tremendously. You can also use a combination of techniques to better understand the behavior of the birds. So this is from a study in Australia, where they used, you know, those little blue tie that you put around the leg of the chicken. So it's a radio frequency identification device. So you monitor every single chicken. They've been using, you know, high-speed cameras, 3D cameras, all those different things, and they analyze the behavior of birds, where they go, when do they feed, when do they take water, and some go outside, others don't go outside, some stay at the ground level, some likes to fly up and down. So really a lot of options. And the more we understand the behavior of the birds, the more we can also design better housing systems so that fits the needs of the birds. Other opportunities, that's the one I was mentioning to you. Some scientists in the United States are trying to study the sound made by the bird, sort of voice recognition, and to see if through listening to birds we can understand what's happening and potentially, you know, disease or stress and so forth. So here's an example I'm going to play for you. I hope it's going to work. And you see the different sounds made by the bird. Here, this bird is scared by a predator. Oh. All right, but you have the other sound. Here is a chicken. The rooster is calling the hen because he found some kind of food. Right, so really different sounds, and they are really working now to try to see if through listening we are able to better understand what's happening in a chicken house. And this is extremely difficult because, as you probably know, if you've been in chicken houses, there's a lot of other noises. If you have automatic feeders, you will have those different type of noises. You have the fan. So it's extremely complex, but there is some great potential here too. Other type of research using artificial intelligence, disease diagnosis, when you have histopathology, and when you do histology, there's a lot of time spent on reading a slide that can easily be done by the machine once you train the machine. So really here, artificial intelligence in many cases is not so much about replacing people, replacing veterinarians, for example, replacing farm managers, but it's helping them do their job in a more efficient manner, right? So here, if you can have a machine that can read slide, that's perfect. Hopefully at some point, we're going to be able to train machines so we can recognize diseases at the very early stage, like incubation, period, so you can really react and potentially, treat the birds even before, when you have only 1 or 2% of the flock that is affected and not 50%. Also, through analysis of the feed ingredient and feed formulation optimization is a possibility. Others, visual grading, so inspection of the eggs just by, and weigh the birds, so the eggs just by camera. So basically, you train a machine, and by taking a picture, the machine is going to tell you that egg is going to be 60 grams, so 58.2 grams, for example. Artificial intelligence also, and certainly a huge application in genetic selection. As you, I don't know if you had some lectures on genetic selection, but it generates a huge amount of data, and individual birds have a lot of information about them. And again, here artificial intelligence can help really analyze better and understand what the data is trying to tell us. Hatchery management, discoveries of new medicine, and so forth, a lot of applications, okay? And then if people think, well, I don't believe artificial intelligence is really there. Well, it's already there, as you know. It's there, used always with digital marketing. And for the poultry business, actually, it's been great value for farmers. So you see the traditional way of marketing in the past, you got the farmers, you got the processors, retailers. The retailers were the one talking to the customers, right? That was the connection. If a farmer wanted to understand the customer, they would have to do, expensive market research to try to understand the consumers. Now, well, this is through digital revolution and the use of those digital technologies. Everybody is connected with everybody. So retailers have still a very important role, but farmers are able through, let's say, Twitter and other type of social media platform can talk to their customers, to their consumers, and they can understand what they are looking for and so forth. So great application, great feature. Okay. Speaking of marketing, so let's look at now the next trend. The third trend here is branding and marketing. Again, in Indonesia, I think just like in a number of countries in Southeast Asia, still a large majority of products of egg and chicken meat are sold as sort of unbranded products, sort of by weights and no fancy, nice packaged product. Same thing in China, for example, China being the largest egg producer and huge egg consumption in China. And for probably more than 90% of the eggs produced are being sold, as you can see here on the left. But recently, the Chinese, the largest egg producers, have created what is called that Wonderful Egg Alliance to try to push more towards the selling of nicely packaged and branded products. And certainly that trend is also very much applicable for the Indonesian market. Again, those two pictures taken from Indonesia from my last trip back in 2017. So a lot of eggs being sold, as you can see here, delivered on the left, on the picture on the left. But you see more now, at least in large urban areas, eggs being sold in those nicely packaged products. And as you think of the new consumers, the new generation, they certainly like better food that is in a nice package, that is clean and so forth, than buying bulk products. So great opportunity to increase the value of the food being sold by farmers through branding. And then the beauty, and here I've got more example about the eggs, is that you can basically, with the same egg, by just changing the packaging, you can target a different segment. So you can see the top left, an egg carton from Colombia, where it's obvious here that the target is young children, right? The one on the bottom left from China, here you are targeting young mothers to show them that those eggs are excellent either for them or for the infant. Good nutrition, it's rich in DHA and EPA and so forth. The egg carton, again, and it could be the same egg, it's just packaged differently. That example from India, where they basically show you with the family, the egg is good for everybody in the family. That's good nutrition. Other ways that you can promote the egg. So on the left here, from Chile, sort of trying to promote fair trade and good return for farmers. And certainly with the COVID-19 pandemic, people have appreciated that they've been able to have food every day. So therefore that a farmer was growing the food and providing the food to them. So really the thing that through COVID-19 pandemic, people have learned more to appreciate where the food is coming from and the hard work of farmers to make sure that food is always present on their table. So that's one way. Another way also learning from the COVID-19 pandemic is the fact that people want, at least in Western countries, they want to buy local food. They want the food regional or from where they live. So this is an example here of eggs produced from a part of France in the mountains. And so, if you're from that region, those eggs are coming from that region. And that's another way to appeal to consumers. Or the other one from Canada here in the center, omega-3 nutrition is more and more important, healthy nutrition and so forth. So again, through promotion of different component of the eggs, you can promote good nutrition and satisfy consumers with the new needs that are derived from the COVID-19 pandemic. Potential also for global trade, more participation through increased production. So you can see here on the left, that's Thailand with the broiler chickens, so the live or the sort of fresh carcass being exported. For Thailand, 2.2% of the world trade, 18% of the production. On the right-hand side, that's the egg. You see Malaysia is playing a huge role in the global trade of shell eggs. 15% of their production is dedicated to export, which is mostly to Singapore. And at the bottom, you can see the results of, in Indonesia, there's no export of either eggs or chicken meat. And potentially, at some point, Indonesia would be able to not only produce for its own population, but also be able to produce and export to other countries. So additional market and opportunities for the sector. Yes, so the egg, typically, the export of shell egg is extremely small. You see only 3% of the global production of eggs is being exported. So most of the eggs are being consumed locally, okay? And Malaysia, I guess, its geography, Malaysia is extremely fortunate to be right next to Singapore, although Indonesia is also not far from Singapore. And Singapore, low production, high consumption, so it's a good market. Okay, another trend that I have here, fifth trend, will be about further processing and opportunities, again, to what I call the new frontier, because it's still underdeveloped markets in many countries, Indonesia, but not just Indonesia, around the world. and huge opportunities to create more products and more business opportunities. So in the region, we certainly, and you probably recognize some of those brands here, very famous in Southeast Asia. For the meat sector, we already have quite a number of big companies in the region, including Indonesia, that will process chickens and sort of create ready-to-eat meals or cut chickens into different chicken parts, so you don't sell the whole carcass, but you sell portions, different portions of the chicken, right? And by doing that, you create tremendous value. You are able to sell more than the price of the whole carcass. And for the egg, this is, and it's certainly less known, so that's why I'm showing you different pictures. This is the type of egg and further egg processed product that you can have. So egg powders, the top left, the hard-boiled egg, then you can see very popular now. It's basically what people do at home. If you have an egg and you put it in boiling water and then you cook it. But in a number of countries, or if you have a restaurant or you have to feed a lot of people, it's a lot of work to boil all those eggs and to peel them. So now you have a lot of companies selling you eggs that are already cooked and peeled. So you have just to open the package and you eat them. On the top right, liquid egg. So here the egg has been broken. So all what you have to do is open the cup here and you pour it on your frying pan and you can cook your product. So again, it's convenience. And with the younger generation of consumers, convenience is certainly a very important element. Make our life easier. This is one option. The bottom left, what is called sort of boil in a bag. So here the liquid egg is in a bag. And if you're in a restaurant and you are going to make scrambled eggs, you just put this bag in boiling water. The egg is being cooked. You take the egg out of you take the bag out of the boiling water, and then you can open the bag and then you pour the scrambled egg on the plate. So it's simpler to do and it creates less mess, less problem for the chef, for the restaurant. And then you have sandwiches, omelets, a lot of different options that are available. Just here again, just to show you the data, there's only about 10% of the global egg production that is being processed. So 90% of the eggs produced are being sold and consumed as shell egg by people, and only 10% are being consumed in the form of those different type of product that I was showing you a minute ago. And if we look at Asia, the number of plants, of factories, if you wish, that process those eggs, you can see that overall in Asia, we have 152 plants. 27 will deal with drying, so make egg powders, and the rest will make those different type of products, liquid eggs. The vast majority of the plants are located in Japan. You see with 82 plants. China, the numbers are growing, has grown tremendously over the past 10 years. It used to be only 2006 was only about 1 to 2 plants, and now you see we have 22 plants. In Indonesia, based on the data that I have, only 1 plant. So again, tremendous opportunities potentially in Indonesia to do more further processing, egg processing, and develop new application of the egg. And again, the idea is that through egg processing, you create many more opportunities for consumers to enjoy eating eggs, right? So it could be like on the left, that picture from Denmark, it's a wrap. So you can basically, it's like a shell, and then you can put some different type of food, and then use that. It's like a sandwich using an egg white wrap. Or you can have snacks, so an egg with some peanuts, some cheese. A global trend, I think, is that people spend less time eating. And They eat at work, they eat while they work, they eat while they drive, and so forth. So you need to provide them with convenience. And also, at least in North America, people like to snack. They like to eat something all the time. So therefore, it's a good opportunity to provide some healthy snacks, and again, simple to make. Okay, another trend is about appealing to the consumers through innovation. And again, let's spend some time on that. So if we look at the global trends for food, and this is from Innova Market Insight. It's a US consulting firm that published every year those different market trends. And for this year, sort of the number one trend was transparency in food, meaning that consumers want to know what's in their food, and not just in terms of the composition, but also if it's animal products, where were the animal raised, how were they raised, what sort of welfare, what food did you give the animals, all those different things, right? So it's very important. Consumers want to know more about their food. The second big trend is all about plant-based products. Again, and you have here an example, that picture, that's meat-free, chickenless tender. So it looks like chicken, but it's not chicken, it's plant-based food, right? The third trend is what we call the food for me. So it's not just in terms of nutrition, but people want to buy food also that match their values, their lifestyle, what they think, what they want, and so forth. So either from the food and also from the company. If you believe in a company, then you're more likely to want to buy food that they produce because it meets your criteria. The fourth trend, which is that one, a direct consequence of the COVID-19 pandemic, is trying to have an overlap. So product that you can find in restaurants, people would also like to be able to find them at in retail stores. For example, if you like KFC food and during the pandemic your local KFC restaurant is closed, you would like to be able to buy KFC food in your local shop. So that's what that overlap means here. And then finally, through the pandemic, unfortunately, we all had to learn a lot more about immune system, antibodies, vaccination, and all those things. So people are more and more interested in food that can help them build strength, build a good immune system. So one of the first trends that I want to talk to you about, it's all around animal welfare, because it is a fast growing concern issues, either from consumers or certainly a lot of NGOs, so non-governmental organizations, are putting the emphasis on bird welfare and making sure that certain conditions are met on farms and so forth. So this is an example of a new concept to raise broiler chickens. It's coming from the Netherlands, and you can see the concept. We have a wide open window, so the birds have a clear visibility on the outside. Also very unusual for broilers, you can see that you have different levels, so the birds can go up and down. You have some straw here, so the chickens can also rest and enjoy different environments. So really, you see more and more of that for broilers. Companies are interested in finding ways to make the life of the broiler more interesting, more stimulating, if you wish, right? And it's a clear demand from consumers in some countries. Same issues with the egg production, where traditionally in Indonesia, most likely I would say 99% of the laying hens are being housed in cages. Back in 2012 in Europe, they say that we have to, the conventional cage is banned, and what is allowed is that furnished cage, which is a slightly larger cage, a cage that has perches so the birds can stand. You see that yellow, those yellow plastic curtains here, that's to create a nesting area where the birds can go to nest and have some more privacy to lay the eggs. So that's the type of cage that is only allowed now in Europe. And later on, I will show you a short video where you can see those different housing systems, okay? And if you look at the trend, what has happened, this is looking at birds in cages. So 2012, almost 10 years ago now, that's when in Europe they say that the regular cage is no longer allowed. And you can see that overall, a lot of those countries have a clear reduction in the number of birds being housed in cages, and they replace either by bird going outside, or birds going in what we call an aviary system, where there's no more cage. It's a big building, and the birds are able to move up and down. Just to give you an idea, again, I don't have specific data for Indonesia, but I expect Indonesia will be very similar to the data that you can see for Mexico, for Russia, for China, in terms of 98% of the chickens will be kept in cages and very few in other systems. But if you look at, for example, all the way at the bottom, the UK, so United Kingdom, you have 42% of the birds that are in cages, but more birds are being kept in free range or organic systems, or birds that is both inside, cage-free, and able to go outside. You know, the UK is the country with the highest number of birds being kept outside now, as a consequence of a number of issues related to welfare or perceived benefit of being outside for the birds. And that push has been driven mostly, as I said, by, you know, consumers. They care without caring so much about animal welfare, but NGOs have been pushing, pressuring companies. So Aldi is a big retailer in Europe, originally from Germany, and you can see this campaign that they had, that an NGO has created, basically implying that if you are selling eggs that are produced from birds kept in cages, this is cruelty, and therefore you are promoting cruelty against animals. It's obviously their view of seeing it. A farmer will tell you that, you know, we put the bird in cages because that way we can best monitor the health, the environment, and so forth. So there is a number of issues, but the NGOs do not care about necessarily about the health and some of the benefit of having bird in cages. They just say cages are bad and you should not you put them in cages, and therefore if you do not, then we will you know, attack you, or we will create bad press for you, and if you're a company on the stock market, your stock price will go down, and this is not good for you. And you know, you see those campaigns everywhere. So you see it in South Africa, in the US, but I was even able to find on a website here one example of a campaign apparently targeting McDonald's in Indonesia, again, to try to stop McDonald's from using eggs that are being produced by hens being in cages. So growing trend, mostly driven by organizations that are very vocal and effective into equating cages to cruelty. And you can see now a number of those large companies have said that they will no longer use eggs that are produced by hens in cages, and only what we call, you know, cage-free systems. And in truth, we need to understand that some of the new housing systems, and again, in a minute, I will show you a short video. Some of these new housing systems have improved the possibility for the birds to express more their natural behavior, like perching, like having truly a nesting area, and so forth, and flying up and down. But at the same time, there's a lot of trade-offs, like the health, the quality of the environment in those cage-free systems. There's certainly more dust. There's more risk of injuries for the birds. So in the end, I think we need to explain to people that there is no currently ideal system. Every system has some advantages and have disadvantages, right? And on top of that, any system, any cage-free system, the birds are much more active, meaning that they eat more. If they eat more, they will have a greater impact on the environment in terms of greenhouse gas emission. Okay, that's what's happening. If you consume more feed, your CO2 emission per kilo of egg is going to be higher. So again, no ideal system. So now, rather than me talking, I'm going to show you a video from this company in Canada. That it's about a 3-minute video. People are speaking, so I won't have to speak, and they show you the different trends, the different housing systems. Okay. So I hope you enjoy this little video.
Oh, lagi Zoom ya?
All right, so I hope you enjoy this video just to show you the different housing systems that are available and some of the advantages and disadvantages of each of those systems. So another, obviously, you know, through innovation, we need to address the consumer needs and demands. So last year, there was an excellent survey being done in terms of trying to understand consumers. You see almost 19,000 consumers, and actually Indonesia was in that survey, representing about 1% of the sampling size, and Asia was quite well represented. So very good insight in terms of what do a consumer think. And if you look at the by generation, so Gen Z, Millennial, Gen X, Baby Boomer, you can see here the percentage of people that are interested in sustainable and environmental responsible products. Millennials, 79%, and Gen Z, 75%. So a high percentage of the consumers are really interested, more and more interested in sustainability, you know, making sure products that they have a good impact on the environment. Other type of response we got, also very interesting, clean products. So here, clean product, the definition would be, you know, safe product. Food safety is a key element. And you can see again, millennials and you know, all the generation above 75% of the consumers are interested in those characteristics. So the survey was extremely interesting because we can see that for food and beverage, so food-related items, if you look at 44%, so the bright blue here on the right, 44% of the consumers are what they called purpose-driven consumers. So we're consumers that are buying food, but with a certain purpose, meaning that they want to make sure that the food they purchase is good for the environment. Okay, they're not value-driven consumers. Would be one that is looking for the cheapest price for the food, right? Brand-driven, you're looking for a specific brand. Product-driven, specific product. Purpose-driven, you want to make sure that the food meets your lifestyle, your objectives, your desires. And when we look at those purpose-driven consumers, quite interestingly, you can see that in fact, 57% of those consumers are even willing to change their habits, their purchasing habits, in order to make sure that they reduce the negative impact on the environment. And 70% are even willing to pay a premium, so pay more money for the same type of food if they know that that food will contribute to reduce the impact, the negative impact on the environment, and contribute to a lesser acceleration of climate change. So it's very comforting, and that's something we need to remember, that we need to discuss more about it, about the impact of egg and chicken meat on the environment and the contribution and what we're doing in order to reduce our impact. And I will show you a few slides later on. So again, if you do research, those are the buying factors for food: price, taste, convenience. That's a given. But then authenticity, sustainability. So any idea, any new product that you need to develop, you need to consider those elements. And sustainability has a huge impact now in the mind of consumers when they want to buy food, and nutrition also, as we discussed before. So one of the very interesting features of the egg is that you can easily, through modifying the diet given to the hen, you can modify the composition of the egg in many different nutrients, right, that have great benefits. So this is a fantastic opportunity for the egg sector, because you can develop a lot of products. And this is how we do it. It's extremely simple. You have a diet, and you add an extra element like vitamins, minerals, different functional ingredients, give to the feed, give to the chicken within 10 to say 3 weeks, 21 days, then that egg is going to be enriched in that specific nutrient. So you create what we call a functional egg. And that transition from the feed to the egg, 20%, it depends. It varies depending on the nutrients. Some will transfer easily to the eggs, others would not easily transfer to the egg. But basically, what happens is that you can modify the nutritional composition of the egg and enhance some of those different elements. And you can see here on this slide how easy so the transfer efficiencies. You look at vitamin A, extremely easy to transfer. So the more you put in the diet of the chicken, the more vitamin A you will find in the egg. On the opposite, if you want to transfer folic acid, for example, which you can also, but you will need to increase more in the chicken feed to get an incremental an impact on the composition of the egg. And you can enrich a lot of different nutrients in the egg. So this is a study done some time ago. You can see different nutrients, different vitamins, the omega-3, alpha-linolenic, and DHA, and you can see the level of enhancement you can have. The level, like lutein, the third one here, multiplied by 6, the omega-3 multiplied by 6 also, and by 3. So you already, through diet fed to the laying hens, you can modify the composition of the egg, and it gives you tremendous opportunity to further enhance the nutritional value of the egg. And those are some of the benefits, for example, of omega-3. You can see the bottom pictures just to remind you that the heart, the eye, and the brain. So efficacy, cardiovascular effect, good for eyesight, and also development of the brain, both in infants and then also later on as you get older. I was very pleased, back my last trip, Indonesia, 2017, see some, in some stores, omega-3 products. So typically, those type of products, production, it's simple. It's the marketing, it's convincing consumers of the value of those products. That is the challenge for the sector, but it's certainly a possibility. Talk more about the egg because it's easier to do for the egg, but you can also, through different formulation fed to the broiler chicken, you can also modify the composition of the meat in those different polyunsaturated fatty acids. So it is possible. It's not as simple as the egg, but it can be done for chicken meat. And here's another example. For example, so different diets fed to the chickens, linseed oil at 1%, 2.5%, or 5%, and you can see the different alpha-linolenic in green and DHA, docosahexaenoic long chain omega-3, sorry. And so you can see a nice effect really of the dose response. So the more in the feed fed the chickens, the higher you will find it in the chicken muscle. And then to again continue with that innovation and the fact that consumers are more and more interested into boosting immune system. Here's a company from Canada that launched last month a new egg ready to drink egg-based product that is promoting the value of this drink to help you support your immune system and so forth. So again, answering clearly a trend that is observed as a post-COVID-19 pandemic observation that people want more food that helps them build a strong immune system. And then the final trend in terms of innovation was about finding your food that you like at retail, in restaurants. You want to find them also in the store, so you can buy them and have them at any time. So opportunities to further develop this business between different companies. And finally, we're almost there, and I appreciate your patience and dedication to listen to me for such a long time in English. So communication, very, very important. Communication, we learned that through the pandemic. And quick slide here just to show you the number of issues and challenges that are being faced by the sector. Certainly cost pressure, we want to produce more for less money. Animal welfare, the well-being of the birds is more and more of a challenge for farmers. Antibiotic-free production is another one. The impact of animal production on the environment, the greenhouse gas emissions, those are big issues that we need to always address. The challenge more and more with the plant-based food. I mean, more and more, it's a growing trend. We have to be careful of that. And then also the bigger challenge is that consumers are more and more from urban areas and less understanding of farming and farming practices and so forth. So therefore, they don't understand. So if you talk to them about putting a bird in a cage, they think it's cruelty. They don't understand the benefit of putting a bird in a cage in terms of disease prevention and preventing aggressivity between birds and so forth. So challenging communication. Here's an example of issues in communication. COVID-19 pandemic started, and in India, there were rumors through social media saying that there was a link between COVID-19 and the consumption of chicken meat and eggs. So consumption dropped tremendously, which is very sad because at a time where consumers were actually needed those food because it was good to build a strong immune system, people were shying away from buying these type of products because somebody had said on social media that there was a link between COVID-19 and poultry products. So we need in the future to be better prepared and be also good communicators. It's sad to see that NGOs, it's always bad news that people are great at communicating, and we always struggle to communicate the truth and good news. A number of other global issues that we see, certainly I've talked earlier about the welfare and housing system, sexing and culling of the male chicks. You probably realize that only female birds will lay the eggs. So in a number of countries, the male chicks are of no commercial value, although they are used to feed zoo animals and other things, as day-old chicks, and that's where they're being used instead of using rats and mice. We use the day-old chicks here. A number of different elements that are important to communicate. Sustainability of the sector is certainly one, and almost done with my slides. Greenhouse gas emissions, you can see here the different sectors, the contribution, and the agriculture sector, it's 14.5%. Overall, that's all inclusive. If you look at animal production, total sectors, we contribute 7 gigaton of CO2 equivalent, and you can see about half is related to the production of feed being given to the animals. Another half, not quite, is the production itself. And then you have very smaller segment. It's what happened after the animals leave the farm. So it's all the processing, distribution channels, and so forth. So, but if you look at the sectors, you can see that the largest contributors of greenhouse gas emissions, it's beef and milk consumption. Egg production, it's only 2.8% of the total animal sector contribution to greenhouse gases. And the poultry meat, it's 5.6. So overall, the poultry and egg sector, it's contributing only 8.4% of the total emissions. So small contributor, which is great. Then we can promote that in terms of we are very fortunate and we need to, that's why we should eat more chicken and eggs and maybe less beef. But sometimes it's difficult, and it's the story gets even better if you look at the emissions per kilogram of protein produced. Again, when we eat food, it's to get energy or to get protein. So here you relate emissions per protein consumed, and you can see both chicken meat and chicken eggs are the lowest, around, say, between 30 and 40 kilos of CO2 equivalent per kilo of protein produced, whereas beef is about 300, so about 10 times more. Okay.
Okay.
And so two
moment.
Yeah. Oh, yeah. 2 more slides.
Okay.
Yeah. And then if we look at, carbon emissions, so one way to explain to consumers is really to show them the impact of some other behavior that they have and how it relates to the consumption of food. For example, if you watch TV, you can see how much emission per hour. If you use your cell phone, you can see the amount of CO2 emission per call. And if you look at basically one egg contributes the same amount of emission as a 2-minute phone call or you watching TV for an hour and 15 minutes. So nobody is stopping you for using your phone. So therefore, NGOs, I don't know why they want us to stop eating eggs, for example. Just one before last slide. Again, importance to communicate with consumers as more and more after the COVID-19 pandemic, they were, eating and cooking more at home. Provide them recipes, ideas to cook. That's a known fact that if you provide ideas to consumers to use your product, they will be eating that product more. So really, it's important to promote and give them ideas. So, and that's my final slide. In conclusion, COVID-19 pandemic, the trends for the poultry and egg sector, it's all going to be linked to being smarter at the way we do things. So we have lots of data, but it's time to, automate the way we use the data, and it's all about smart data. It's also all about using automation and all those latest technologies to make our life easier and so work smarter. And then finally, looking at the food supply chain, it's all about sustainability and having a smart value chain and communicating those different elements to the consumers. And with that, I will thank you very much for your attention. If you have any question, I'm happy to try to answer them. And I did send my slides just before the lecture, so they should be available to you if you want to read them or you want to keep them or whatever. Okay?
Okay. Thank you very much, Dr. Vincent. I believe now we will have some question from the participant. So someone already raised hand, Jordi Aditya Prameswara. Excuse me, Dr. Vincent, could you please stop share?
Oh, yeah, sorry about that. Here we go.
Yeah. Someone, Jordi Aditya.
Yes, sir. Do you hear me?
Yes.
Yep.
Okay. Thank you, Dr. Vincent. Very, very interesting presentation. And I have a question. In Indonesia, before it, nice to meet you, Dr. Vincent.
Yep.
And I have a question about egg.
Yeah.
In my country, Indonesia, a few years ago, there is an issue about fake egg.
Yes.
Fake egg. And I try to ask it with my lectures, and my lecturer said it's impossible to make a fake egg. And my question.
It is possible. I've seen it also in China. That was unbelievable.
Oh, possible, huh?
Well, yeah, I mean, it looks like an egg, right? But you cannot eat it. It's lots of different chemicals. It's crazy to me. When I saw that in China, I could not believe it first, and even that it could be cost efficient, that you could make those things that looks like an egg in a cheaper way than having a chicken that lay the egg, right?
Oh,
but
okay.
It is. I've seen it. I heard about it. But I've not seen it. I heard about it. I've seen videos from China. but it has no nutritional value. Okay.
Oh, okay.
It's just a scam to make money, and no.
But there is a possibility in the future, we use that product. We talk about the growth technologies right now, availability technologies right now to make it, and is there a possibility in the future we use that product, artificial egg?
So yeah, so now it's not a fake egg. It's not something that looks like an egg, but it's not an egg. Now it's using modern technology to grow an egg, maybe in a petri dish, right?
Okay.
That I don't know already of any much research in that field. What we see is actually people trying to replicate the characteristic of an egg using only plant-based elements. Okay, and that is happening. But obviously, then it's a liquid product, right? It's not something that looks like an egg or anything like that. But you're gonna buy a bottle that it's going to tell you it's a vegetarian egg, for example, or whatever they want to call it. And it's only plant-based elements that are trying to mimic the color, the taste, the texture, the characteristic of the egg. Okay.
Okay.
And it's quite interesting. Some of those products, they have like the egg, it's only one ingredient, it's the egg, and those plant-based egg, it's like 15, 16 different molecules to try to mimic, to replicate the egg.
Okay.
All right.
Okay. Thank you, sir. Thank you. I got it. I get it.
For you?
It's clear. It's clear for me. Thank you, sir. Nice to meet you.
Okay.
Pleasure.
Okay. And then maybe going to the next question is in the chat room from Ina Rofi. The question for Dr. Vincent first. What is the basis chicken layer in free cage? What about productivity for commercial production?
So in cage versus non-cage?
In free cage, non-cage.
Cage free.
Yes, cage free.
Okay, yeah. So cage-free birds typically are going to consume more feed. So you have maybe, say, 15-20% more feed consumption. And you will typically have also maybe 5 to 8% less egg production. Okay, so basically, cage-free birds, you will probably have slightly lower production and more feed consumption. So your cost of production is going to go higher. Okay. And in addition to that, the contribution of greenhouse gas emissions is going to be higher because more feed, more emissions.
Okay. One final question for you, also from the chat room. My question is, what is the biggest challenge when we are going to move from conventional cages to catch free based on animal welfare? Because in Indonesia, about 264 million layer birds still use the batteral cages. What is the biggest challenge transforming?
Well, and again, to me, Indonesia does not necessarily have to move from batteries, from cages to cage-free, okay? I personally believe that the new cage system, the enriched cage that I show you, the one with the perches, with the nest box and so forth, is probably a better compromise in terms of improved welfare and still safety to the birds, worker safety, sustainability, protection in the environment, and so forth. So really, if you have the chance, and maybe through communication, through a number of things, those enriched cage system or furnished cage systems are probably a better alternative. Now, if Indonesia were at some point forced to go cage free, then obviously this is challenging. There's a lot of management issues that are involved, and the best for farmers will be to try on a small scale first to learn how to modify their management practices, because it is much more complicated and challenging to manage bird in a cage-free environment. There is more dust. There is usually higher ammonia level compared to a cage system.
Okay. I'm not sure about how I will say to the closing remarks, because your explanation during the lecture today is cover all the, I think, the aspect of eggs. Okay. So before I end this session, I will invite Professor Asep to give you the certificate from our department. Professor Asep?
Yeah. Yeah, Pak Reza. Hello, Dr. Vincent. It's nice to see you.
Yes,
live
pleasure.
today. Yeah. What time now in Canada?
Yes, it's almost 6 o'clock in the morning.
Oh, we are really thanks and appreciate to your effort to give us the lecture and presentation. Very nice today.
It's okay. I hope next time.
Sorry, actually we want. Actually, we are very pleasure to welcome you. Yeah, to welcome you after the COVID-19 is decreased and free, and then we are really to see you here in Bogor. Have you visited in Bogor?
I did once, many years ago. Yeah.
So thanks a lot on behalf of the committee, and also you are very kind and contact me, and also Ibu Maria here, the contact for the first contact, and we would like to thanks for giving us the lecture in our summer course program. We are really happy and enjoy hear your lecture and presentation. Very nice presentation, and also update information related the trend of the poultry industry development in pandemic COVID-19. So we are really thanks, and here maybe please accept the e-certificate, and also we will send you a letter by email, and then again. So we would like to thanks, and we hope we can furthermore to collaborate with you in the future, and also in the coming letter. Thank you.
Well, thank you. I just want to thank all of you organizing committee for the invitation, and I would like to congratulate also the students, and because listening to a lecture in English, I guess most of the program is in English, right? So this is not easy. I mean, I know, because English is not my first language either, but I had 30 years of practice, so it's good. But listening to a lecture in a foreign language, it's not easy. So congratulations to them, and all the best, and looking forward to next year's session, if you want. All right, all the best.
Okay.
Yeah, thank you.
Terima kasih.
Terima kasih kembali.
Very nice word.
Excuse me, Dr. Vincent. I believe Dr. Maria would like to say hello.
Oh, okay. Yes.
Dr. Maria, Ibu Maria.
Ya, Pak Resta.
Oh, please, time is yours.
Morning and good afternoon, Dr. Vincent. Yeah, afternoon, and in Canada still early in the morning.
Yeah, it's time to get up now at 6.
On behalf of the World Poultry Science Association Indonesia branch, I would like to express our gratitude and appreciation for your great support to our program in collaboration with Department of Animal Production and Technology and Faculty of Animal Science in conducting this summer course. And please also receive our apology to bother you early in the morning to give presentation. So it's very great to learn from your side from your experience regarding to the poultry development during COVID-19 pandemic and also after that. And I learned a lot from your presentation. And we are very exciting to for further collaboration with you, maybe next year or in the near future. Thank you again, Dr. Vincent. Have a nice day in Canada, and thank you so much.
Pleasure. A great program that you have put in place, and I'm looking forward to someday meet you in person.
Okay.
And again, congratulations to the students.
Okay. Thank you very much, Dr. Vincent. From me, the moderator, and from our department, we like to say thank you very much for just giving the lecture for hours. There's an update lecture, and also thank you very much. And sorry to wake you up earlier in the morning.
It's okay. So, no worries. That's part of the deal.
Okay. So I have some hope that maybe after the pandemic, sometimes in the future, we all together here, we can meet you in the person, maybe in the Bogor someday. Okay?
Yes.
Okay.
Oh,
okay.
bye.
And see you. Bye.
Thank you. Pleasure.
Okay.
Bye-bye.
Okay. Bye. Okay. Now the time is giving to the committee, please. Pak Arifino, maybe Pak Arman. Okay.
Yes.
Alhamdulillah Pak, di akhir-akhirnya itu sudah Bayu kumpul itu. Waduh, bikinmu melayang jadinya. Ya, that's true. Excuse me, for group 16 in where the back room for our group 16.
Yes, hello, sir. Good afternoon. Hello, can you hear me?
Yeah, it's good.
It's clear. Okay. By the way, my name is Ellen from the Philippines, and I would like to ask regarding the presentation for tomorrow. Is it from the group one? We will start from the group one, or is it a random?
Maybe you can share the technique.
Please, Ellen.
Yes. Can you hear me?
Yes. Yeah.
Okay.
Yeah.
Okay.
Okay. So what
So,
is your
sir,
question? Sorry.
I would like to ask regarding the presentation for tomorrow. Is it from we will start from the group one, or is it a random? Just like you need to pick a number.
Okay, yeah. So for the techniques tomorrow, Pak Dimas, maybe also you can add. We will divide the group, yeah, into break room, yeah, and then for the presentation, we will give you opportunity actually. From the less number of the group until the big, yeah, the big number of the group, yeah. But actually, since you are ready, yeah, since you are ready, for presentation tomorrow. Yeah, maybe not make a random. Yeah, we are not make a random actually. Yeah, Pak Dimas. Yeah, but we will give the schedule. Yeah, the schedule according to the group. Yeah, we will map like. But of course, you to attend. Yeah, you have to attend in all the group presentation. I mean, since maybe like okay, so now it's not my turn presentation. Yeah, okay, so maybe I'm not joined that. Yeah, but you have to see. Yeah, you have to see the group. Yeah, the group, all the group. Yeah, in the presentation, in the break, like that. Yeah, Mas Dimas. Yeah. Yeah.
All right, sir. So thank you so much.
Yeah, like that. Yeah, like that. Yeah, we have, but I think it's also good opportunity from you. It's also okay. Maybe we can accept also maybe from the committee, since you are ready to present. Yeah, maybe we can like in the group. Yeah, in the group. When you already enter the break room, yeah, for example, in group A, 1, 2, 3, 6, 7, 8, 9. So you have the 9, and then maybe if you want, yeah, to present not from 1, 3, 5, 7 or something like that. Yeah, so maybe we can like random. Yeah, so maybe the committee will make the paper, maybe 9 paper, and then we will. Okay, so the first group maybe could be, for example, group 11. Yeah, and then second something like that. I think it's maybe it's more fair for all the participants. Maybe it's good also maybe for us maybe. Yeah, to make a randomized tomorrow. Okay, it's okay. Yeah, Pak Dimas, yeah, maybe we can notice. Yeah, we can notice for tomorrow, and then we can randomize the number. So it means all participant, all group, yeah, ready to give the presentation at the time. Yeah, okay. Thank you, Ellen. Any question? Any other question? Yeah. Okay. I think everything maybe is clear. Yeah. So tomorrow we can start at 1 o'clock. Yeah. Please on time again. Yeah. So we have parallel 2 break room with a large group. Yeah. 17. So we have we hope we can finish. Yeah. Before 5 o'clock, because closing, yeah, closing ceremony, yeah, for closing the summer course program. Yeah, at least official tomorrow the summer course will be closed. Yeah, otherwise maybe we have yeah, in 2 or and 3rd of August. Yeah, international student conference like that. Yeah. Pak Dimas, any other information from the committee? So it's already actually, yeah. Okay, yeah, yeah. Okay, so if everything is fine, and then good luck for you, and then see you tomorrow for the presentation. So you can free if you want to make maybe communication with the group. So I think it's better, yeah, because the summer course that we can hope from you, not only yeah, hearing the lecture and then hearing our presentation of excursion, but the important thing you can collaborate, network together with the group. Yeah, so please feel free if you want to make a, for example, maybe exercise for the presentation, so you can arrange. Yeah, you can arrange, since we have time. Yeah, tomorrow on the afternoon, maybe you have a time around maybe 30 minutes, yeah, for the exercise. Maybe I think it's also good. Yeah. Okay. So thank you very much, and then have a nice evening, and then see you tomorrow.